then we have been taught at that time, when you look at cell-mediated immunity, humoral immunity, and how is it impacted, what is the regulatory process, and how does the hypothalamic pituitary, the adrenal axis and others influence the immune system. So you have every cell talks to each other, every system talks to each other. It's not individually, they work alone. In an integrative physiology, it is always the whole integrative human being is considered as one system in which immunology plays a very important role. Now, as I said, immunization. Now, how do we improve further to many diseases? Opportunity. Now, students, at your stage itself, you should think which is the area I'm going to now make a dent, not just make a degree, get a degree. That is the first step. But where I am going to make a dent, whether it's tuberculosis, as our professor is doing, Nishit is doing tuberculosis, or it is organ transplantation with Professor Uma is doing. So you have to now look at what are the areas in which you are going to make a dent and prepare yourself for that. Equip yourself and with all your knowledge, skills, and also the commitment to achieve something unique. Because everybody gets a degree, gets a job, and then make a family, we disappear. But some people are remembered today. So you should be remembered. Each one of you should be a success story to give something to the society. So your degree, getting a degree is the first step, but go beyond that. What Amitians will do is this. They will make a mark in life, and all of you will make a mark in life. Then when you look at transplants, that is the Madam Uma's field, in fact. So look at the mechanisms that how do you prevent rejection of the transplanted organs, systems, tissues. Now, so many the organ transplantation is coming up now, new fields. So where the immune system immunomodulation becomes very vital. So look at those fields as a great opportunity. And then there are many traditional systems in which there are good immunomodulators. In fact, when I was at DRDO, I worked on an immunomodulator coming from Sankapushpi, Amla. These are all naturally available. Amla, all of you may be taking it also. So there are sources from your traditional knowledge which can help in immunomodulation. So which we should look at. Don't look at only synthetic molecules, drugs, and so on. Look at your own traditional systems. How do you practice yoga and improve your immunity? Your immunity in terms of cell-mediated humoral immunity. I have done some research in that area. So go beyond your textbook and look at the horizon, widen your horizon. Look at the traditional knowledge also as one of the areas in which India can make a mark, because you have so much of wealth of traditional knowledge available. Explore them, understand them, and adopt them in the modern concept in terms of doing a randomized, double-blind, placebo-controlled studies and so on. Put it in the scientific pedestal. There's another opportunity for immunologists to develop immunomodulators, adjuvants for vaccines. We did some work on adjuvants for vaccine using traditional system in DRDO, Defense Institute of Physiology, where I worked. So this is another area which we should look at. The other one, which is immunotherapy for cancer. You have a strong cancer group here, Dr. B.C. Das, who is the father of cancer cervix, the HPV. So he is here and he is heading a strong group on cancer. So how we can use immunotherapy for cancer treatment? So there's a lot of opportunities coming up now. Then look at the immunotherapy for cancer. Then you just organized a few days ago, maybe a few weeks ago, neuroimmunology. Because there are many diseases which are caused by either autoimmune disease or it caused by neurodegenerative disorder leading to the immune dysfunctions. So both ways, it talks both ways. It becomes causative factor or sometimes it becomes the effect on the system. So understand this neuroimmunology. Neurological coordinates influencing the immunological system and immune system impacting your neural systems. Opportunities. There are many pathological conditions which we can cite where this dialogue takes place and making the pathological conditions. The next one, which I have worked in an area, psychoneuroimmunology, which is very important. The psychological profile, your anxiety, stress, happiness, can influence your immune system through neurohumoral axis. The immune system can be impacted. The very interesting experiment I did on breast cancer patients with Bangalore Cancer Research Foundation and with Defense Institute of Physiology, Defense Institute of Psychological Research, and CCMB Hyderabad. It's a major program which we did where we looked at the perceived stress of the patients. How does it influence the immune profile and the prognosis of the patients, the recovery to the treatment? So what we did, we selected 60 patients of breast cancer. Also, we selected 60 spouses, the caregivers and spouses. We measured their perceived stress using the psychological questionnaires, anxiety, state anxiety, trait anxiety. So we took the profile, perceived stress, quantified way. Then we looked at their cortisol, catecholamines, adrenaline, noradrenaline, and then we also looked at the immune profile, CD4, CD8, NK cell, PHA activity. All this, we looked at the immune system. So psychological, neurohumoral, and then we also looked at sympathetic, parasympathetic activity to indicate the sympathetic arousal in these patients and spouses. So we looked at a system rather than only patient-centric. That's why we involve the spouses and caregivers. Then we looked at the strategy, coping strategy of the patients. Every patient adopts her own coping style, or if it's a spouse's, his own coping style. So we looked at the coping style. Then we did a multivariate analysis to find out the correlation. Which of the coping style helps in prognosis? Which of the coping style which leads to the progression of the disease? And then how does the perceived stress influence the immune system? So like this, there are opportunities. Then we introduce psychotherapy, which is essential. Besides doing a surgical removal, radiotherapy, chemotherapy, and then you have to give psychotherapy, the counseling, and also yoga, meditation. So we have given that message that psychotherapy has to be a part of psycho-oncology, which is now introduced. So such kind of opportunities available for you. So this is the time when you can explore such possibilities. Now, finally, to give the message, India is now in a very vibrant position today. So your role, our role, have increased. With when you looked at Azadi Ka Amrut Mahotsav, tremendous achievements in every field, not only health sector, agriculture, a begging bowl India was at the time of independence is now a breadbasket. We couldn't feed 30 crore people. Now 1.4 billion people, 140 crore people, we are not only able to feed, but we can give food to other countries also. That is transformation, food security, health security. And in terms of space, SLV, ASLV, PSLV, GSLV, Lunar Mission, Mars Orbital Mission, and Gaganyaan, many more, Aditya coming up. So, what I want to say is we have achieved so much in 75 years, which is not a big period for a nation. We have achieved. Now, we have to now look at Amrit Kal. Next 25 years, what we are going to do, as our Prime Minister says, that we have to be a developed nation. Why wait for 2047? Soon, in another 5-10 years, we should become. For which we people have a big role. Now we have to go beyond and go beyond the call of your duty. And then prepare yourself as students, learn more, think differently, disruptive technologies, processes, medical management, and also new processes should emerge from India, from you. So that is why Amrit's call, my clarion call to all the professors, students, is that look beyond and start thinking that your role is not 1.4 billion people, it is 7 billion people. Look at the 7 billion people. So you have to prepare yourself to assume that role. So this is what Amity is doing to all of you, that realize that, and I'm sure you'll excel in your life. And two things I want to convey finally. One is mission synergy. founder president Dr. Ashok Chauhan has given three missions which I like. One of them is Mission Connect, the other is Mission Synergy, the third one is the mission outcome. So, Mission Connect, now I was connecting with them, with the aims, what we can do, with Drishti, what we can do. So, this is Mission Connect. Then, in the process, what I can give it to them, their mission, synergy. What they can give Amity's growth, synergy. So, next thing you should look at how do you network among yourselves with other institutions, connect and bring synergy for both. Third one is the mission outcome. Whatever you do, whether if you are listening to this lecture, there must be a sustainable outcome, not that you have learned something and finished. How do you apply this? Ruminate that, and then it should lead to something else in a sustainable way. So, whatever you do, have a sustainable outcome. So, I would expect from this meeting also what is the sustainable outcome for our students, for our faculties, for our guests. So, this would be the final message I want to give you. Celebrate successes and be happy in life, then you can do more things. Keep a mind cool, happy always. Then happy mind can be innovative, creative, disruptive. All that will come in a happy mind. A happy mind. So this is what is my experience, which I thought I should share on this International Day of Immunology. My best wishes. Thanks once again, Devender Tour. We are very fortunate to have such brilliant leaders here, like Devender, who is an achiever, who is again very quiet worker. But when I saw his profile, I must salute to him. You know, the kind of connect visibility he has at international community. He is doing an international project, which you may not know. International project he is doing. Let's clap for Devender too. So such people, we are fortunate. Amity is blessed to have such people. And once again, thank my guests who are come here from Dishti as well as All India Institute of Medical Sciences. We want to connect with you. We want to bring synergy in your endeavors and our endeavors, and we will have a sustainable, fruitful outcome for both the organizations. My blessings to all of you, my children.
Thank you, sir, for your words of wisdom. Our next speaker is Professor Nishit Agarwal, sir. Dr. Nishit Agarwal obtained his doctoral degree in biochemistry from the Delhi University South Campus in 2004. Subsequently, he joined Johns Hopkins University, USA for his postdoctoral research training. His group established for the first time the role of pathogen-derived cyclic AMP in mycobacterial virulence. Dr. Agarwal identified an adenylate cyclase of Mycobacterium tuberculosis, which was critical for intracellular survival of the tuberculosis pathogen. After completing his postdoctoral research work, Dr. Agarwal joined the Translational Health Science and Technology Institute in 2010, where he is currently working as a professor. Dr. Agarwal has authored 30 research publications in peer-reviewed journals. He is the principal investigator and co-investigator of multiple extramural and collaborative research projects and has supervised more than 30 project fellows and short-term trainees. We invite you, sir, to enlighten us about tuberculosis research in the 21st century, status of diagnosis, therapeutics, and vaccines.
So good afternoon, everyone. First of all, I would like to thank Dr. Devinder and Dr. Narayan Rishi for inviting me. So this is my first visit to this university, which is not very far from where I live. I live in Amrapali in Noida. But I've heard a lot about this place, but never got a chance to visit this. But I must say that I'm really excited to be here. So, as rightly said by Dr. Selvamurthy, that we are celebrating Immunology Day to commemorate all those major achievements which we have achieved and which are helping us. COVID has given us some snapshot of this generation about how immunology and other sectors of the health can be helpful for the for humankind. So when Dr. Devinder asked me to give a lecture on this day, I was a little perplexed because I don't work on immunology, neither am an immunologist. But since I work on tuberculosis, I thought that tuberculosis is a disease which, unlike other pathogen, is not because of any particular toxin. It's rather because of the perturbation of immune response. And I found that this can be a good area to discuss on this auspicious day of World Immunology Day. So that's why I kept the title very general, which is tuberculosis research in the 21st century. What are the status of diagnosis, therapeutics, and vaccines? So in today's presentation, I would give a brief overview of the history of tuberculosis. What are the current advancements in the tuberculosis diagnosis, and what are the vaccines in pipeline? Subsequently, I will touch upon the history of TB chemotherapy and the current therapy of tuberculosis, followed by identification of new drug targets in the post-genomic era and the screening of small molecule inhibitors. So the last two areas, I'll give some overview of our work. So what is a tuberculosis? As you can see here, it's one disease, but it has many names. If you go back to the history, it used to be referred by different names, such as phthisis in ancient Greece, tabes in ancient Rome, and shakafet in ancient Hebrew. So it is believed to have evolved 3 million years ago, but we have the written evidence of it being evolved in almost 10,000 years ago in humans. So the molecular studies have revealed its presence in 1/3 of Egyptian mummies. So just imagine that how much was the prevalence at that time. And historical reference to TB in Egypt are in a medical papyrus dated 1550 BC. So it's quite ancient disease. And the spinal tuberculosis, also known as Pott's disease, has also been detected in these sculptures. So for a long time, we did not have any treatment, any understanding of the TB before 1700, when it was called as white plague, okay, because of the paleness of the patients. So people were not having any idea what is going on. Then Johann Scholin coined the term tuberculosis for the first time in 1834. Okay, so despite his nomenclature, it was still called as consumption because it used to consume people's life. And, it was called the captain of all those diseases of the death. So it was very, very devastating situation. Then a French military surgeon demonstrated its infectious nature, and he, for the first time, did an experiment, what we call the experiment, by inoculating rabbit with a small amount of fluid coming from the autopsy of the individual died of TB, and found that the animal is also getting the disease. So this was the first indication of it being contagious. Subsequently, in the similar time, around 1867, Klebs tried to isolate TB bacillus, and he tried hard to culture these bacteria and using different media, egg white, etc., and then subsequently infected guinea pigs, and which they found that they are catching infection. However, for the first time, Robert Koch, who is also called as father of tuberculosis, so at that time, he identified, isolated, and cultivated bacilli in animal serum. And finally, he reproduced the disease by inoculating these bacillus into laboratory animals. So for those of us who work on microbiology, we still follow Koch's postulates, which I've just given a glimpse of here. So what this Koch's postulate is, he gave several principles to basically attribute an organism with a disease. So the first principle he gave was that a pathogen should be present in a diseased animal or the diseased individual. Now, in the present age, I will say that there are exceptions for all these principles, but by and large, we still follow in the microbiology labs that if we have to confer any pathogen with a disease, then we have to find its presence in the diseased individual. Subsequently, second principle he gave was that these organisms should be culturable. Now, why did I say exceptions? Because now we know that there are very lot of organisms which are fastidious in nature, so they cannot grow in on a simple medium. So, like for regular practices, you can grow E. coli like organisms, like all the sepsis organisms, you can grow on LB plate. Okay, but you cannot grow Mycobacterium on LB. So that's a whole lot of history and research associated with, just like Dr. Selvamurthy mentioned about adjuvants for the medium development. So there were scientists called Middlebrook who was first succeeded in designing media which can be synthetic medium, which we are still using for culturing mycobacteria. And that's why the TB research is not only time consuming because of the slow growth of the pathogen, but it's also very expensive. Because unlike LB medium, your Middlebrook medium is almost 10 times higher in the cost. So just imagine that how difficult it is to manage. So the third principle he gave was that if you infect these pathogens which you have recovered from the diseased individuals into a healthy individual, it should lead to disease again and cause the death. Okay. And then the fourth principle is that you could again retrieve these pathogen from the individuals or the animals whom you have infected. So this was the principle given by Robert Koch in 1882 after identifying the pathogenic nature of the bug causing tuberculosis. And he presented this paper on 24th March. And since a long time, we are celebrating the World TB Day on this day. And we have just celebrated at our institution a month ago, where we have commemorated a conference on World TB Day. So coming back to the life cycle, why it is so difficult? I mean, why do we talk at all about TB? We see that, in the daily life, you keep getting infection, and particularly in India, the infection, bacterial or viral infection is not that uncommon, Almost every year you have these episodes of 4 or 5 infection cases. But why does it so, difficult for TB pathogen to eradicate and cure TB? Because of its pathogenic life cycle. So if you can see here, it starts from aerosol inhalation. So we do not know. I mean, I've been hearing it. It is often very embarrassing also when you travel to the Western world that, oh, I went to India so and so years. After coming back, I became PPD positive, which is a sign of latent TB infection. So it has been attributed that because of crowding and inhalation of the pathogen from the infected individuals who are asymptomatic, we catch infection. So if I can give the statistics as per the World Health Organization, almost 25% people sitting in this hall might be having TB pathogen in their body asymptomatically. So this is why it is so concerning and so difficult to treat. So what happens that when the pathogen comes into the body, but it encounters the infection, the body encounters the infection, and I'm talking about the pulmonary TB, what is most prevalent. There are a whole lot of extra pulmonary TB which infect other organs than the lung, which we do not talk much about. Now people have started realizing about that with more advancement in the science. So they are taken up by specialized cells, what are called the macrophages. So these are the primary defense cells. If you talk about the immunology, these are the first. You can say that these are the border territory, army, which picks all the invaders. So majority of the infections are taken care by these macrophages, and then they bring, because of the elicitation of immune response, lot of other, immune cells in the vicinity. And at the end, the body tries to basically form a solid caged-like structure, what we call the granuloma. So this is granulomatous lesion is often present in the body, undetected in the body for many years, which does not give you any harm. You are happy, bacteria are happy. So for a long time, it used to be considered that the granuloma formation is host protective. I'm not showing data here, but when I went for the postdoctoral research, so we did work on the cyclic AMP and we broke that myth that the granuloma are host protective. We gave that analogy that they are bacterial protective. So it's a synergy which we have just talked about between the pathogen and the host. So you keep us happy, I will keep you happy. I will not harm you, you don't harm me. So we showed that the TNF alpha, which is the primary protective factor in the active cases, is actually driving this granuloma formation. And the evidences were also supportive at that time when people were taking anti-TNF therapy for rheumatoid arthritis. And they, in a country like America, where the prevalence of TB is not very high, many of them turned into active TB cases because of the latent TB infection. So that then started realizing that the role of TNF alpha in basically maintaining the latency of bacterium and preventing them to become active. Another problem with TB is the different stages, which are very, very narrowly defined. There are no such clear distinction. I've highlighted here 4 different stages. So the first case, either you are innately immune, which is shown here, and do not catch infection, or you have caught the infection and without you realizing, your body has actually fought with it and eradicated that infection from your body. So in both the cases, while if you do not catch infection, your lungs are clean. But if you have caught the infection and cleared it, you see some scar in the lung, which comes in the X-ray. So those who are basically Indian clinicians are very much used to seeing this kind of scenario because of the high prevalence. And based on that, the advices are given. So no such therapy is recommended for both the cases where you have not got any infection, obviously, but even when you have got the infection and you have been treated, body has taken care of it. But for some reason, when this converts into latency, this is a major gap currently, that what drives latency, and your body is unable to basically get rid of the pathogen. How do we know that whether we have the latent infection or not? I'll come to that when I will talk about the diagnostics. But the latent TB, as I said, is very prevalent as per the WHO estimate. Overall, globally, on an average, 1 person out of 4 contains the TB pathogen in the form of latent bacterium. While in a country like India, this percentage must be very, very high. Why it is concerning? Because the latent TB infection is the infection which is often found to be responsible for conversion from the latency to the active disease, especially in the old individuals. And why it is so prevalent? Again, if you see, you might have noticed in your family, who have the elderly members in the family, they have some, nowadays this pain in the, legs and other spinal joints, etc., are very common in the old age, especially. And in the earlier time, people used to take all the Ayurvedic medicine, yoga, etc. But now everybody goes to the clinician, and clinician does what? Gives you the painkiller. Okay, and the painkillers are basically, or sometimes they are the immunosuppressors. So what has been observed that these painkillers, immunosuppressors, are also contributing in breaking that, barrier of the latency and conversion into the active disease. That is one reason. Secondly, if you read about the literature, news, etc., you see our population is getting very much diabetic. So diabetes is also increasing alarmingly in our population. And this is called the comorbidity. That again perturbs your immune barrier and convert the latent disease into the active disease. So that's why latency is very important to manage. Unfortunately, we are doing very less to basically address the latency. And obviously, from the subclinical to the active disease, the window is very, very small. Okay. So I'll come to the diagnosis aspect. What are the various approaches for diagnosis of tuberculosis? So again, giving you some history, because I love this. This history is very important because that often encourages you to work on what you are working on. So when Robert Koch basically identified the pathogen, he thought that if he will give the bacterial extract, it can act as a cure, because at that time it was very common that, for some unknown immunity kind of thing to be generated, but it was found ineffective to cure the TB. But it was found that in the period of 1907 to 1909 by Clemens, that you can actually use this to detect the TB infection, what he called the latent TB infection. But the first time, basically, Charles Montew. So now, even in regard of this scientist, we call this a Montew test. What was done was that a small quantity of the bacterial extract was injected beneath the skin by a needle, and to basically look for the hypersensitive response. At that time, it was not known what is happening, but what all they were expecting was some induration. And even now, this is basically it's one of our protocols in the biosafety committee, that if you have to work on tuberculosis, you have to get the Montew test done. And if the induration is more than 5 millimeter, you are considered as latent TB individual. Now, if this is the case in United States, in their occupational health, you are immediately recommended to take the isoniazid therapy. So this is the preventive therapy. In India, because we are living in the high prevalent region, we get rid of this, saying that okay, we might be having latent TB because its burden is very, very high. So, but by 1930, so just a gap of 30 years, there was not a very standardized protocol for making this purified protein derivative, what we call the PPD. So for the first time, this American Florence Siebert basically designed a procedure, a standard operating procedure, to formulate the PPD formation, and which we are following now. Now, one of the major disadvantages with the PPD was, as I said, we are living in a high pandemic region, that it is unable, and we are vaccinated, by the way. I will come to that also. In India, it's one of the mission in the Dhanush program, whereby just after the birth, you are given BCG vaccination. So because of the cross reactivity, because hypersensitivity is nothing, it's just recognition of the immunogen and antigen. So because of the cross reactivity, basically it is difficult to say that whether it is because of the BCG vaccination or because of the TB, latent TB infection, that you are getting that hypersensitive response. So almost 10-15 years ago, this advancement came, the advanced diagnostics test came, called the interferon gamma release assay, IGRA test. And these tests basically are two types. One is a T-spot TB and another is a quantiferon TB gold, which were simultaneously started being used to detect real TB infection from discriminating from the BCG. And all these advancements happened because of the knowing the genome sequence of the mycobacterial pathogen, which came in 1998. Recently, the nucleic acid amplification test, what we call the gene expert, has also been invented, which is used for measuring the drug resistance in TB. So I'll come to that also in subsequent slides. So what is the T-spot TB? Because this is the immunology conference and this pertains to interferon gamma release assay, so this is very important for all of you to know what is the T-spot TB. So basically, when we take the whole blood, there are mixed type of cells. Okay, so one first step is that you have to isolate the PBMCs, what we call the peripheral blood mononuclear cells. So the PBMCs have different cell types, including your T cells, B cells, etc. So what you do is that you take a 96 well plate, which is coated with anti-interferon gamma antibodies. Okay, and you basically, now here there's a advancement that you basically sensitize your cells with the two of the antigens called the ESAT6 and the culture filtrate protein 10, CFP10, which are basically absent in the vaccine strain BCG. Okay, it is only present in the virulent organism. So basically, your T cells, which have seen prior infection, will show a robust response. And what you do is that you wash them off after binding, and then you develop signals by using the secondary antibodies. Typical of ELISA, but it is different from the ELISA that you have spotted the antibodies against interferon gamma. So how is the readout? As shown here, if you are having a positive result, is something, the control is nil, where there is no antibodies. Okay, The ESAT6 is the panel A, which is the first of one of the two antigens. You see a lot of spots here, but nothing in the negative. Then you have another antigen, CFP10, which is again give you a spotting. And then the positive control, chemokine is taken basically to use a non-specific. So just to tell that the reaction has worked. So on the similar principle, this another company, Coagen, has basically formulated the quantiferon TB Gold. Here, what you do is that you take so there you have to separate the cells. Here they claim that you don't have to separate the cells. You can use the direct blood. The principle is same, that you are sensitizing them with the TB antigen, pathogens antigen, and the mitogen is taken as a control. And then you incubate for 24 hours. Then you basically measure the release of interferon gamma by using the ELISA. Now, when these two tests were invented several years ago, there were very malpractices started happening in India. So because of that, now Indian government has stopped this test in India. Why? As I said, that none of these tests can distinguish between the latent TB infection and the active TB infection. And these tests cost you 4,000 to 5,000 rupees per test. A lot of money were being made by the private hospitals from the poor patients, because TB is the disease of the poor, basically. They cannot afford, and the exchequer was coming to the government. So there's a very limited now, under basically advisory has been released for the use of these IGRA tests. I named the NAT what we call the basically nucleic acid amplification test. So this is again a very useful invention in the current series of the tests, and we have seen the benefit of the nucleic acid amplification test during the COVID time, when everybody was rushing for the RT-PCR. Now the RT-PCR has become the household name, and the days are not far when it will become a word in the dictionary by Oxford also. So this is such a power of pandemic which we have seen in the recent time. So the gene expert, this is called the cartridge-based nucleic acid amplification test, CBNAT. So what is the CBNAT? Basically, it is basically a very simplified test where you take the sputum sample and then you put on the cartridge, and the cartridge has been given all the automation. So at the end, it extracts the genomic DNA of the pathogen, and with a very sophisticated probe, it amplifies and tells you whether the pathogen is actually there or not, or whether it is positive for the drug resistance. Nowadays, it is largely used for rifampicin resistance or not. So the major advantage of the CBNAT is that its robustness and its ease of using. The only difficult part is that it is not there in the tertiary care centers. So it requires in the referral centers, because it requires a lot of infrastructure and the expertise. Recently, our lab has also added one more test on the NAT, what is called the CARP assay. So it's basically nothing. It's just that the CRISPR-Cas associated reverse transcription RT-PCR based test. So the principle here is that when you have the wild type and the mutant form of the DNA, and you design your probe in such a way that can detect either of the wild type or the mutant, so what will happen that because of the Cas9 endonuclease activity, based on the matching sequence, you will see a cut in the DNA amplicon. And when you will do the RT-PCR, if the DNA is fragmented, your RT-PCR CT cycles will increase. And based on the difference in CT, you can say that whether the resistance is there or not. So this is the result I'm showing, that the level of detection is very, very high. It can actually detect up to 0.01 femtogram amount of the DNA. And the CT differences are such a big differences, 11 versus 15. So almost 4 or 5 CT differences you can see based on what kind of guide sequences you are using, matching or not matching with the mutant sequence. So coming to the vaccine, the second part of my talk. What is the vaccine basically? So as your immunology students, you must be knowing that vaccine is basically to protect us from the infection, future infection, okay? So what about the TB? Because the TB, which has been so old, it has been living with us for years, thousands of years. So, but there was no vaccine till 1921. This is a very unfortunate part. And in the 1900th century, the two great scientists, Albert Calmet and Jean-Marie Camille Guérin, these are French scientists, they started working on this. And I was fortunate when I was in Johns Hopkins to be in the group of a great legendary French scientist called Jacques Grosset, who was part of this team actually, and he used to basically proudly telling us, narrating his experience with the vaccine development, that he used to say that my job was only when I was employed was only to subculture the bacteria. So I was not sure that what kind of job they are taking and paying me so high. So his job was only to subculture bacteria. Every day he used to go and subculture bacteria. But often it turned out that, the subculturing has resulted into an attenuated strain, what the world is now calling it as a BCG, in the honor of Bacillus calmette-guerin. Okay, so the whole lot of development has happened. I will not go into that, but I will only say that BCG is, helping us a lot in India, basically, to prevent our children to get, protected from the disease TB. So that is why BCG is mandatory, has been put in the mission in the Dhanush program, given free of cost in the all the government set up healthcare system. But where BCG fails, then one can say that all of us are vaccinated. We are vaccinated from the chicken pox. We are vaccinated from the MMR. We are not seeing those diseases. But then why we are talking so much about TB? The reason is simple, that BCG fails in adult population. Okay, so the huge trial was conducted in the Chingalput. It's a famous Chingalput trial, which was done for many years, because the effect of vaccine, that is another problem in the vaccine for TB. The effect is seen after several decades, whether it is protective or not. So this test was done for almost 20 years in all more than 260,000 patients. And what was the result? You see the result. Protection is none. Okay, so the variability, if you can see, is 0% to 80%. But one observation was that it is still protective against the TB meningitis, which is very prevalent in infants in the up to the 5 years of the age, and that is why, to prevent them from the infection of the TB meningitis, BCG is mandatory in the newborn. So now what progress we have made. So I've listed several of the vaccines. They are not visible here. You don't need to worry about that. So you can just see that these are the phase trials. So the two vaccines which are listed here, I'll come to them also. Then the advanced stages of the trial. So the one of that is called the VPM 1002. So I've just highlighted some of the features associated with the VPM 1002. This is an attenuated vaccine derivative of the BCG, which is now in trial in India also. So we are part of this trial, and the huge trial is going on, the VPM 1002. It is basically a derivative of BCG, in which what they have done was that they have removed a gene called the urease, okay, and they have introduced another gene called the listeriolysin. So what the listeriolysin does is that it breaks the membrane of the phagosome, yet let the bacteria be in the phagolysosome, and it basically maintains the acidic pH of the phagolysosome, so that the antigen presentation happens by both MHC class 1 and MHC class 2. So basically, the VPM 1002 trial is going on. The results are yet to be awaited, but the world is looking at the this vaccine, because they have high hopes that this will have a more broader immunity compared to BCG. So this data shows in animals they have done, and shows that it's the bacterial burden on the left y-axis is a logs scales basically, which clearly shows a significant reduction by VPM 1002 compared to BCG in animals. So this vaccine is also under trial in India, and we will see its result in next few years. Another is the MTB-VAC, which is again in the advanced stage of trial. So MTB-VAC is the vaccine which is the first ever vaccine derived from the pathogenic strain of TB. A lot of debates were going on whether we should consider attenuated TB strain as a vaccine or not, because the BCG is derived from the M. bovis, which causes TB-like symptoms, disease in the bovine population. It is not, we humans are immune to M. bovis. So BCG is relatively very safe. But MTB-VAC is derived from MTB, where again two genes have been deleted. So these are shown here. MTB VAC. So the first of these genes is the basically 4P. It's a 4PR is a basically sensor and response regulator. So what they have shown is that the 4P is responsible for the release of many of the antigens which you can see here, but they are present in this mutant. And another gene which was deleted from this vaccine was the FAD26. So if you look at the architecture of the bacteria, Mycobacterium tuberculosis, the architecture is very unique in terms of from compared to other bacteria, that it contains a lot of lipid-rich cell wall. Okay, so one of those lipid, and several of them have been attributed as the virulence factors. So one of them, called the thioceryl dimycosidosate (TDIM), is also found to be what because of that it forms the cord-like structure. So we call the cord factor. Okay, so if you ever get a chance to look at the morphology of the bacterium on the solid agar plate, you'll see them serpentine-like structure. It's like not like other bacteria like E. coli if you have grown. They look very shiny. This is not like that. It's very rough because of lipid and a corded structure, what we call the cord factor. And that cord factor is the major virulence factor. If you just remove the cord factor from the bacteria, they will become attenuated. So they removed that FAD26 gene also from this. And this was done at the University of Zaragoza in Spain. And this vaccine is also approved by TB Alliance for vaccine human trial, and this is currently under trial. So we are hoping that these two vaccines will definitely give us more power to fight with this disease as far as the vaccine is concerned. This is the data they have shown in animals that the protection is much more by the MTB VAC, which is in the center, or at par with BCG. In two different experiments they have done. The experiment 1, it is slightly better. Even in the experiment 2, in the lung CFUs, in the spleen, the effect was not that great, but at least it is at par with BCG. And therefore, something which is as good as BCG or better than BCG is approved for trial in humans. Okay. Now, coming to the third aspect of the disease, what is the treatment options we have for tuberculosis? This is very important because all of the two things which I've told you, obviously disease is very important to understand the prevalence. Vaccine is important for long-term protection. But what if you have failed at these two levels? Then you need to have the antibiotics, okay? So again, there were a lot of myths associated with it. I've just highlighted some of those. So in early 1800s, it used to be considered as vampire panics. So people used to believe that people dying with TB come back as vampires, So you have to basically dig their grave, do some rituals so that you can get rid of infection in the locality. They never realized that this infection is coming from the air, actually. So, and then to that extent that their part of the exhumed body, it's basically used as a cure. So they used to give that, extract of that as a cure, but obviously it is not going to do anything rather than creating more problem. People in the age of royal kingdom, used to be having queues outside the kings for the royal touch, believing that this will again heal their misery. But again, it did not work. Lot of other therapies, even sometimes you see even nowadays, cod liver oil, vinegar, this and that. But nothing used to work. What used to work, what Dr. Selva Murthy rightly pointed out, to keep yourself happy, get good oxygen, get good food, basically in a nutshell, improve your immunity. So having realized that when the vaccine, when the drug was not there, people built this sanatorium. Okay, so the sanatorium, even they do exist now. So sanatoria are nothing. They used to be very good, peaceful area, isolated places in the hilly regions, basically, where these TB patients used to be sent. If you watch some old movies, you will see them, TB patients being sent to the sanatoria, and some of them getting healed, many of them at least living for much longer time period than otherwise. So Edward Livingston Trudeau basically built the second sanatorium, and he himself died because of TB. And Trudeau is given a high regard in America because he developed the sanatorium for where the TB patients used to be sent. And another idea was to basically, by that time, people have realized that it is very contagious. So you have to keep them away, just like, in the present day, we have seen for the COVID. So any disease related to airborne infection, they have to be treated like that. But then first treatment of TB came in basically 1950s when the Salman Waxman developed first ever basically vaccine called streptomycin, which was used against TB. So this paper was published in the British Medical Journal in 1948, where the first ever trial of TB drug conducted on humans, and the results were very impressive. As I don't know whether it's visible or not, so it was found that a considerable improvement was observed in the 51% group compared to those control groups where the success was only 8%. So this was very encouraging, but this encouragement did not last long, and soon they published another paper that streptomycin resistance started happening. So patients who were given streptomycin treatment soon came back now with the streptomycin resistance. And just imagine that they did not have any other antibiotic at that time. Serendipitously, this is not visible. This isoniazid is mentioned here. So this drug was there not for any infectious disease, but this was there for developed for some other purpose. They started doing trial with this drug, and then they found that this was very effective and published another paper almost 10 years after that, the first ever trial conducted with the drug regimen where rifampicin and this isoniazid and streptomycin was given, and they found that there was around 70% improvement compared to what was found with the streptomycin alone. Now, what are the current therapy we have? So I've listed here in 3 blocks. This taken from NIH CDC. So the very regular treatment which we have currently are the combination of 4 drugs, by the way. It's the rifampicin, isoniazid, pyrazinamide, and ethambutol. And they are taken for 6 months, not less than 6 months, sometimes more than 6 months, based on your basically performance. But despite these 4 drugs, we often see resistance to primarily 2 drugs, what we call the rifampicin and isoniazid. And whenever clinicians see the resistance to these 2 drugs, the pathogen is attributed as MDR, multi-drug resistant. And that is where now it has become mandatory to basically diagnose drug resistance before onset of the treatment using the technology called the Gene Expert. So this CBNet is very helpful in basically detecting the drug resistance, mainly the rifampicin resistance. And it has been seen that when the patients have the rifampicin resistance, majority of them have also resistance to isoniazid. Okay, so for multi-drug therapy, you can see treatment. You can see a lot of these drugs. I'm not going to ask you to read all of them, but you can just see that how much development we have made, and all these have come in just last 20-25 years, not less before that. So we have many such drugs which are like which act on the protein synthesis, which act on your DNA replication, which act on the your ATP synthesis, respiratory metabolism, etc. And then we have the extensive drug resistance also, by the way. So the bacteria are so clever. Virtually, they develop resistance to all the drugs which are given, and what we call the extensive drug resistance. So here it is mentioned. So for multi-drug resistant, the therapy goes for 2 years. It's very, very difficult to treat. And every day you have to basically get the injectable drug, So last year only the first oral regimen has been developed for the treatment of multi-drug therapy. Just imagine that for 2 years, every day you are taking drug in your vein. So what kind of life you are living? And a few years ago, I was reading about a journalist personal, experience in the Times of India published on the World TB Day. 80% deaf. So she is totally deaf and lost eyesight. The body is totally malfunctioned basically. So you're just living your life like callous. You're no more functional. You're basically life quality of life is getting deteriorated because of the TB. So there are several new drugs have been developed in the recent time. And if you think about all these drugs' characteristics, they don't do anything. They just touch upon the same areas like, the cell wall biosynthesis. Here's your central dogma, which is the information pathway, DNA replication, transcription, and the protein synthesis, and the ATP metabolism. The drug ATP synthase inhibitor is the recent addition in the form of bedaquiline, what we call. And government is so careful because of the resistance. The bedaquiline is not recommended as the over-the-counter drug. It is only given under the supervision, and in case of multi-drug resistance. So we are still fighting to basically get the drug for which the resistance is unable to be created, or at least new drugs, so that we can treat the difficult to treat drug resistant infection. And it is not a joke. Because of that, in 2021, the situation is like that. Globally, we have almost 11 million TB cases, and out of them, 500,000 cases, we are seeing the drug resistance. Unfortunately, because of the poor diagnosis and prognosis, only a fraction of those, almost 36%, are getting treatment for their drug resistance. And out of those who are getting treatment, you see, only 60% we were actually able to cure the disease. So that means almost 80% of the drug resistant cases we are losing every year because of the not having proper treatment. Okay. So the question comes now that how to identify new drugs. All right. Now, this question might be burning in your mind, that how scientists basically develop new drugs. Okay. It's not an overnight development. It needs lot of systematic studies. And the first step in that is the identification of the drug target. So this is the genome sequence which came in 1998. Just imagine that must be wondering why we were not able to develop so many drugs as now we have seen in last 20, 30 years, because of the no information of the bacterial genome. We did not have any clue at that time. And that is why a simple genome sequencing paper came in Nature, because it was so fascinating to see the nature of the bacterial genome, which is 66% GC content. So it's not a joke. Like, unlike other organisms like E. coli or other bacteria, which contains 40% GC richness, here the GC content is 66%. And why GC content is so matter of concern? Because, as we all know, that GC bonds are linked by 3 hydrogen bonds compared to 80 by 2, and therefore the replication, any such event which requires separation of the DNA strand, requires more energy to break the 3 GC bond compared to 280 bond. And that is why the bacterial machineries overall become slower. Okay. So after long time and many of research, people have identified 500 genes which they could not basically disrupt, and they claimed those genes as essential. So the first criteria in basically attributing any target as a drug target is that this gene should be essential. If you want to kill your enemy, you want to kill in the head, not on the limb. Okay. So what are those genes which are acting like heart, lung, head for the bacterium? So these are the 500 genes which they found that putatively essential. Okay. But problem is that how to identify their essentiality? These are just prediction. Okay. Until we do not identify, we may consider them as essential and say we have invested 10, 15 years considering their nature as essential, which is actually non-essential. And we have invested a lot of money, developed a lot of molecules against them, but it turned out that neither that molecule nor that gene is essential for this. So that is what the major question. Okay. So for that, we have to follow, to attribute a function of a gene, we have to follow again a Cox postulate, but now it is called the molecular Cox postulate. What is the molecular Cox postulate? The phenotype or the property under the investigation of the gene should be associated with the pathogenic nature. So if a gene you call this a virulent or required for the essentiality, it should be an inherent quality of that gene. Okay, it should not be present in the avirulent organism. When you delete that gene, that property should be lost. And when you introduce that gene, that property should be regained, what we call the complementation. So phenotype and the genotype association is very well defined by Stanley-Falco in the form of molecular Cox postulate. So this is the snapshot of how the development of the genomic perturbation has taken place since 1990 about Mycobacterium tuberculosis. I will not go into detail of it, but what our lab has done basically. So since here up to 2012, all the technologies which we have for genome manipulation were based on the homologous recombination. The efficiency was very, very poor. So what our lab was, started doing in 2012, 2013 was that we started indulging ourselves into the CRISPR-Cas approach. And at that time, it was very, very novice. I mean, much paper were there on the giving genome editing application of the CRISPR-Cas. So I just have highlighted here how it works. So it's basically a large protein called the Cas9. And it has the property to recognize a a small DNA piece of sequence, what we call the Cas9 handle. Okay, so what we did was that we attached a small sequence with a Cas9 handle, which is specific to the target sequence. So now what will happen that because of this target sequence and the Cas9 chimera, it will bring the Cas9 at that site which we want to basically disrupt. Okay. Another sequence which basically provides a specificity is called the protospacer adjacent motif. So it cannot cut until unless it does not see the protospacer adjacent motif. And because of the combination of these two, it will cause a basically double-stranded break. And that you can easily see on the gel. This is the lab-generated gel where you have incubated with Cas9 only, no cleavage, but you have just when you have added the guide RNA, which basically is a chimera of the handle and the target sequence, now you see cleavage of the DNA. But the question was that how to inactivate genes which are presumably essential, because if you will cleave that gene, bacteria will not survive. They are essential for the cell. Okay. So for that, we have designed this new approach, what we call the dCas9. So we made two simple mutations at the 10th position and the 848th position. These are the active site mutations. And we have converted aspartate and the histidine into alanine. So now what happens? Every property remains same except it does not cut the DNA. It is nucleus inactive, dead. So what happens that once it is there, it will silently just create a roadblock for movement of RNA polymerase. So the RNA polymerase will not be able to transcribe that gene. And if the gene is essential, basically the movement, and this you have to do conditionally. Okay, so you have to let the cell grow and then slowly you induce the expression of the dCas9, the guide RNA, etc. So slowly you will see cells getting dying. I'll show some picture of that also. I hope I have here. So this is how it looks. The moment you're giving the inducer here, either the cell is not growing or growing weaker compared to the control. So these are the two genes taken. One is the cell division and another is the respiratory metabolism. Both of them are not letting bacteria grow. So we have used this further. We have multiplexed this approach. At the same time, you can basically silence multiple genes. Also, I've shown about the CARP assay, which we have developed for diagnosis. So this is the list of the genes which we have created mutants in our library. So ours is the only institution in India having such a vast collection of MTB mutants. Virtually, we have touched upon all the essential genes. We have collection of almost 700 mutants in TB strain, touching upon all as such genes which can be attributed essential. And that is helping us to basically reveal and understand which of them are actually essential for bacterial growth in vitro or not. So in just a few couple of more slides, I will show some data related to the genes which I have highlighted here. It's called the CLIP machinery. So what is the CLIP machinery? The casinolytic proteases. These are the chambered proteases constituting two different protease subunits, which form the tetradecamer complex formation. It's just like you can say that you have seen the tandoor, So they form a tandoor kind of structure, and on the top of it, ATPase is attached, what is called the unfoldase. So that provides the specificity, because if the protease is there, cell also wants to protect its own protein to not to get lysed. Okay, so what happens that simple mechanism is given where the cognate substrate it either attaches with the adapter protein or has some signature sequence called the digrons. Basically, the aim is to be recognized by this ATPase, which can recognize this as a prospective substrate, and then basically turns it into brings it into a protease chamber in that tandoor, in which the protein is basically hydrolyzed into small pieces. Okay, so in E. coli or many other organisms, this machinery is not essential, by the way. But in mycobacteria, it was predicted essential. Why it was essential? It was not known. So this terminology of maintaining the protein homeostasis is called the proteostasis. So I'm just shown here that if you treat the cells with a bacteriostatic drug called the chloramphenicol, it basically blocks the movement of ribosome. So translation is momentarily blocked. Okay, it's a reversible binding. That's why it is bacteriostatic. And when you chase them, you start seeing their, them being silenced after a while. So if you just profile the protein, you'll see that different time scale, different proteins are vanishing with a different rate. So that is what is called the half-life of the protein. So every protein has a certain half-life. What determines their half-life? That is something which is very, very interesting to read, and that is where our lab is mainly involved into. So this is the picture I was talking about. So these unfoldases, when you silence their expression, just imagine the growth. There's no growth actually. So these cultures are like this. The moment you introduce the induction of the Cas9 and the guide sequence against these genes, the cultures become watery. So they are not growing. So that indicates that they're essential. Okay. And when we repeated this chloramphenicol experiment in the CLIP mutant, we found many proteins which are relatively more stable, suggesting that they are dependent on this CLIP machinery for their degradation. Okay, so we did a simple experiment to identify what those proteins are. And at the end, we found by all the bioinformatics studies, we have come across a very interesting observation that many of those proteins which are dependent on the casinolytic protease for their maintenance carry a disordered regions at the N and C terminus. So this was very interesting because disordered sequences are not given very much importance by the structural biologists because they are unable to be folded properly. But we found that they are the characteristic signature. So we picked one such protein called the HSP20. Shown here is its level in the mutant, but in the wild type, in the another unfoldase, there was no expression. We found that it's a very highly oligomer, and we show that it's associated with the unfoldase by a BLI octet. When we model this, we found that it's a C-terminus region is hanging, and this basically C-terminal region gave us a clue that this is the digron for this particular protein. When we removed the C-terminus, there was no interaction, and when we express this protein without C-terminus, now we can see its expression in the wild type cells also. So that means the bacterial protease machinery is unable to detect the protein prospective substrate lacking the C-terminus region. So this is the basically in summary we can say that the anywhere the disordered region or either on the single end can be recognized. But what about the proteins which have the disordered region in the middle or they don't have any disordered region at all? That is what we are trying to study at present. Okay, so just to summarize that what we have shown that in basically CRISPR-Cas mechanism, we can identify the drug target, and I've shown one such protein which is very essential for in vitro growth. Now, if we have to basically target that with the drug as a drug target, what is more important is that it should also be essential for bacterial growth and virulence in the animal model. So to address that, what we have done was we basically infected mice with two different strains. One is the control and one is the mutant. And we measured the bacterial load in the lungs at different time points. So here is the experiment where we have introduced the inducer of the Cas9 so that now it can suppress the expression of gene in bacteria which are residing in the animal's lung, okay, or in the spleen. So what is happening? So in the control strain where there is no guide sequence, nothing is happening because of that. But in the case of mutant, the moment we are giving inducer, the CFUs started going down. So that's indicated that basically this gene is not only essential for in vitro growth, but is also required for maintaining its virulence. And this is the lung picture. There's a lot of granular matrix structure which I just talked about are there in the control. But in the inducer treated where the Cas9 based suppression of gene is happening, we are not seeing that much of lung pathology also. Overall, this indicates that this gene is essential for maintaining MTP virulence. Okay, so once we have ascertained that okay, this gene is essential, we started hunting for its small molecules which can target this gene and inhibit its activity so that we can develop that molecule as a new drug. Okay, so what we did was that with the help of our collaborators, one of them is the synthetic chemist, medicinal chemist in BITS Pilani. We've started a screening of molecules, developed a very elegant assay in the plate, and this is the compounds which we identified, 4 or 5 compounds, which are also shown here, which were showing more than 80% inhibitory activity against the that particular ATPase. So that indicated that these molecules are basically doing something related to the activity of that particular gene or protein, which is essential for bacteria. So when we check their effect on the bacterial growth, out of many, we found one molecule, what is called the NSC10010, which is developed by the National Cancer Institute in the United States, was giving minimum inhibitory concentration of less than 1 micromolar. So anything less than 1 micromolar is acceptable as a treatment option. So we call them HIT molecule. We also showed that it interacts with the C1 ATPases. So we did the dose dependency, and what we found that it's up to 40 micromolar. It is basically inhibiting the 50% activity of the enzyme. and its dissociation constant, which tells us that what is the affinity of this particular drug with the protein is somewhere in the 2.8 micromolar range. So then we started doing the structure activity relationship. So what is the structure of this molecule? So this structure indicates, when we look back into the structure, it's 2,6-methoxy 2-methylquinoline ring. So basically, this is the quinoline ring containing methoxy group at the 6th position, and they are linked by a linker called the bisamine linker. Okay, so we also showed that this inhibits the degradation ability of the entire protein complex. So to understand, to develop that into a drug, because this is not the final drug, basically, we started working on some, on this molecule. And the first experiment we did was that we varied the chain length, which are linking the two quinoline ring. And we found that there's no much effect as such. So our conclusion was from that there's no effect of the length. But what about the methoxy group? So when we, this was the methoxy 2-methoxy group. So when we removed that methoxy group, the inhibition has been reduced by 10-fold from 74% to 7%. And that was the case across all the 3 derivatives, indicating that the methoxy group is very, very important for this compound to inhibit the ATPase activity of CLIP-C1. Then we again started asking the role of these bisquinoline ring. So we removed one of these 2 rings, and we found that the MIC has increased. So now more drug is required, means its potency has decreased. So that indicated that both the quinoline rings are important. Okay, so bisquinoline chemotype is very, very essential for it. Then we started identifying the effect of the linker region, because this is very flexible molecule. That's why we could not dock it by usually what people do, because it's very flexible. So we thought, okay, let's introduce a bulky region in that, but we did not see much change. So that indicated that linker's flexibility is not the case, at least as far as the function of this particular effect of this drug is concerned. Okay, but when we removed a methyl group from here and added a electron withdrawing group, we did not see any inhibition. So the potency of this drug is completely lost, and therefore that indicated that electron withdrawing group at the 2nd position and the is very detrimental. So methyl group at the 2nd position and the methoxy group at the 6th position are the key of this molecule. Subsequently, lastly we tried changing this bisquinoline chemotype with the bisquinazoline, which is a similar analog, but when we again saw loss of the activity. So that indicated again that the bisquinoline is very, very important for this drug. So overall, if I can summarize from our, from my last 45 minutes talk, is that basically diagnosis of TB is very, very challenging. This is what we can take, home the message from today's lecture from mine. The diagnosis is still very challenging. BCG is unable to protect, so we need new vaccine, definitely. How we can reduce the burden? So if we can timely diagnose, we can start the treatment at the right time. Most of the diseases are basically we fail to treat because the patients reach to the clinician at a very later stage. Clinicians are not God, so they also need the timely, intervention. So if we can diagnose well, we can definitely increase the TB treatment. If we can prevent the population by giving a superior vaccine, which is very effective in adult population, then again we can manage the disease. And if we have the good drugs which can act on the drug-resistant TB cases, then again we can manage. And because of the CRISPR-I tool, we have been able to identify genes which can be used as the potential drug targets. And using one such protein, CLIP-C1, we have shown that yes, we can screen molecules, identify molecules, and understand the mechanism of their action. And we are hopeful that this scaffold we can take forward to develop a new drug in near future. So with this, I will end my talk of the day by thanking all those who are involved in this work. So Dr. Sandeep Sundaryal at BITS Pilani, he's a medicinal chemist, and he has been involved in this work throughout the beginning on the CLIP-C1 inhibitor development. Dr. Ramandeep Singh, who is my colleague and has a long time friend, has been involved in the screening of compounds. The PhD student, Dr. Ajitesh Lungje, who is a medicine, who's also a medical doctor also, and he has just finished his PhD. He was involved in the characterization of CLIP protease, and Linus was involved in the CARP assay development. and the technical support by all the technical officers. So thank you very much, and I will be happy to take your question. Thank you.
Thank you, sir, for this wonderful talk. Students, any questions?
Hello, sir. My name is Soma Mazumdar. You said, sir, in Gene Expert used for TB detection, but Gene Expert only use sputum sample. So it's only going to detect the pulmonary TB. So what about the bone TB, CNS TB? How do they detect?
So let me first clarify that Gene Expert is not used, not recommended for TB detection, because the simple reason that it is based on the nucleic acid amplification, and the often bacteria are dead, yet they leave their genomic material in that site for some time. So you can falsely identify a person who has cleared the infection based on the some piece of the genomic DNA left as a TB infected. So Gene Expert is mainly used for detection of the drug resistance. Okay. Coming to your question, that what about the extrapulmonary and the CNS TB? Very good question, I will say. We don't have any definite test for them. All we do is that we do detect by the hypersensitivity based PPD test, Montu test. But as I mentioned, we are living in a high pandemic region where even the latent infection is very, very high. So that is why it is difficult to say that sometimes, like a lot of time it happens, and my students go for BSL-3 working, so they go for the PPD test and they see huge induration, hypersensitivity, huge induration, like the whole arm is swollen. So that is kind of scary, but they don't have any symptom. So they have the latent infection. So therefore, for all the extrapulmonary TB cases, it is only on the case-to-case basis. Our clinicians are smart actually. So sometimes the symptoms itself tell them. So in India, what happens usually? When you come to the clinic with any disease, the first treatment is given that दवा ले लो टीबी की. ओके, आफ्टर 15 days डॉक्टर साहब कुछ फायदा नहीं हो रहा, ड्रग रेजिस्टेंस टेस्ट कराओ. So this is a very routine, and then oftentimes it has found that person has cancer, he's taken 25 days of the medicine. Yeah, somebody comes with a acute pain, arthritis है, यूरिक एसिड नॉर्मल है, पेन किलर से कोई फायदा नहीं हो रहा, बायोप्सी करी गई तो पता चला कि भर भर के बैक्टीरिया है. So the hallmark is the culture. So what you do is that you do the smear, you create a smear and look for the acid-fast bacilli, what's called the AFB staining. And oftentimes it has been seen that a lot of studies also people do, clinicians do in AIMS kind of hospital where they do also research that when you take the sample, pleural sample, or from the spine fluid, you basically do the biopsy for the AFB staining also. And sometimes you see and correlate that. Same for the knee. So joint TB is very, very common. Bone TB in that case, joint TB is very common. So all that goes by, but hallmark is culture. But no such definite test, unfortunately, we have. Yeah.
Hello, sir. So, sir, my question was, can invasive fungal infection can make the person to be more susceptible to this infection?
See, fungal infection as such has no direct relation with TB infection. Fungal infection has a relation with the immune status of the cell.
Sorry, sir. So, if we are talking about the infection related to Cryptococcus neoformans, then?
To my understanding and knowledge, I don't have any such evidence which suggests that the prevalence is increased or the susceptibility is increased with a fungal infection. But yes, any kind of these are all called the comorbid status. That is another thing. Like when we do the animal experiments, we do a very clean animal experiment. Okay, but when we talk about the real-time scenario, we are seeing many other co-infection. So what is the effect of co-infection on the TB or any disease per se? COVID, we have seen everything. So what is the effect? But at the end, you will see there is no direct link actually. Everything goes by the immune status. So we know that, for example, HIV. Why TB has become so important, by the way? TB has become very, very important in the Western world because of the HIV. Because the HIV was the first infection where people have identified that being caught in the TB patient. Reverse. TB is the first infection called in HIV patient. So basically later on it was identified that CD4 plays a major role and providing the protective immunity. But still, only the CD4 is not 100% responsible factor for preventing us from the TB. It's this T cell CD8 mediated T cell response is also equally important. So I say that any infection which perturbs your Th1 Th2 immunity will definitely make you to look for its effect on the TB infection. But so far, in my knowledge, I don't have any such case.
I was asking this question because I was wondering that for the vaccine protection against the same disease, Cryptococcus neoformans, maybe the vaccine can also get the person to be protected from the infection of Mycobacterium tuberculosis.
Well, definitely. But as I said, people have tried multiple. So another approach people have followed is because BCG has always been used as the vaccine, platform. So they've tried to express many antigens in BCG itself. Okay, and none of them they found showing better result than BCG. So that is a $1 million question, that what protects us, even if we talk about the younger population, from TB infection because of the BCG vaccination. We still do not know with clarity that what is so protective power in BCG which allows us to be protected for certain years from the TB infection. So that itself is a separate area to study, that what are the factors which are responsible for, because of the long duration, people just spend their entire career, and nobody wants to publish only single papers in their entire career on that. So yeah.
Thank you, sir.
So not such studies have been done. Yeah.
So students, any other question? Okay, sir, I have a small question.
Yes.
You talked about different diagnostic techniques like NAT and then the other version of it. So when we decide that we have to go for these expensive tests, because we already have a tuberculin test, so when to decide that we should go for these expensive tests?
So I will tell you that TB is mostly prevalent. If you just consider the entire global burden of TB, largely it comes from South Asian countries, that too from India and China. Whereas in the Western world, TB is not that prevalent. To that extent that BCG is not mandatory in United States. In fact, they don't take, Americans don't take BCG vaccination. So there it is very easy, where the disease burden is low. It is very easy to detect by tests like IGRA, interferon gamma release assay, because you do not see environmental bacteria, what we call the non-tuberculous mycobacteria. That prevalence is also very high. In India, what happens that basically, as I said, you are asymptomatic. If you go for the PPD testing, the Montu test, no wonder that it comes positive for you. Okay, now you will ask, a lot of queries come to me. Doctor ne bata diya mujhe TB hai. First thing is that whether you have symptoms or not. So the thing which decides, which is the decisive factor, it's mostly subjective. There are certain very, very clear symptoms of the disease. The first symptom, which is very clear, is the loss of appetite, loss of weight. If you are not planning anything and constantly your weight is, you're not gaining weight, number one. Persistent cough for more than 2 weeks. If you don't have any allergy or anything and you are coughing and throwing sputum, loss of weight. Third is the night sweating and mild temperature. So these 3 or 4 criteria, if you go to the doctor ki, अब मुझे कुछ समझ नहीं आ रहा, I mean, I'm coughing and the intensity of cough is increasing, then what doctor will do is that the first thing he will go for it, okay, go for the TB test. Now, there are 2 kind of tests. I did not mention the other one. PPD is the primary indicator, but as I said that it also falsely identify the latent TB. When you throw the sputum, they do the culturing, okay? So the culture takes 1 month. Nowadays, they started doing the liquid, this culturing, what is called the midget medium. There also takes 2 weeks. So based on that culturing, culturing is the gold standard. So when you develop any diagnostic test, that is the criteria what is called the point of care testing, POC testing. There's the criteria that it should match with the gold standard. So your gold standard is basically presence of bacterium, which is culturable on the plate. But that takes a lot of time. In 1 month period is very critical. So doctor क्या बोलता है? Okay, start the therapy. All right. So for the therapy starting, also there's a caveat. You do not know whether that therapy is going to work on you or not, because if you have the drug resistance. So oftentimes gene expert is not suggested because that's a molecular test. Before COVID, that also used to be very expensive. So your question is that when to decide which test has to be followed. There's no such benchmark. It's only your symptoms, clinician's expertise, okay, correlation, and the response to the treatment. I remember recently a bad incident happened with one of my colleagues. So his sister was coughing for a long time, and when he brought her to the clinician, they basically put her on the anti-TB therapy for 1 month, and it did not work at all. They also went for all the other tests. Nothing came out. Finally, they found that she has a lung cancer. So that is something which is we have to be very, very careful. The only way to do is kind of, you can say, initial measures that you are giving the treatment, the therapy. Now that is also coming with a price. It has been seen that once the therapy has been started, you should not stop intermittently, because that adds the fuel to the fire, leading to a lot of resistance, because we have the latent bacteria. And that is why it is up to the clinician to decide based on all those parameters, looking at the stage of the individual, whether or not to start with the therapy. But here our clinicians are so overwhelmed with the patient. 2 minutes mein bas therapy le and then they are done with it. Yeah.
So thank you, sir.
Thank you very much. Thanks.
And I also take this opportunity to welcome Dr. Taruna. You can, Dr. Taruna Madan, she's on the screen. So welcome, Dr. Taruna. And
hello, Devendra, can you hear me?
yes, you are audible, ma'am.
Grateful, grateful. First of all, Devendra, my sincere apologies to you all at MIT Institute of Virology and Immunology that I could not be there in person. And today only I actually traveled back from Delhi to Mumbai because of some important office work. Otherwise, I would have stayed back. And next time, Devendra, whenever I am in Delhi, I'll inform you and I'll meet you all in person.
Thank you. We look forward to you here in Amity University.
Sure. And a disclaimer, Devendra has, mentioned me as Professor Taruna Madan, but I am not a professor. I'm only a scientist with Indian Council of Medical Research, and I'm heading the Department of Innate Immunity. And to tell you all the importance of immunology training, I am not an immunologist by education, but I actually became an immunologist by attending to lectures by experts and reading it personally, because I had taken up a challenge during my research which involved the understanding of immunology. And that's how I became an immunologist. I'm a pharmacy person. I did my B Pharm, M Pharm, and then my PhD was also in pharmacy, but it had a component where immunology was involved. And to tell you very frankly, Nishit Professor Nishit talked about his new novel therapies for tuberculosis and novel strategies for therapy for tuberculosis. And Nishit was there in our continuing medical education program come international conference at Miranda House. So we at IIS are now aiming to reach every nook and corner of the country so that we can at least provide a basic understanding of immunology to all our life sciences students. And this is very, very integral. And I'm sure, and the questions your students were asking to Nishit, I would like to, convey my apologies to Professor Uma Kanga that I could not attend her talk in person as I was in the flight. But the questions that students asked to Nishit were great. Both the questions were very great. And Nishit did provide them some clues, and I'll provide you some more because I have been working in this area of differential diagnosis of fungal infection, aspergillosis, and tuberculosis. So that was the PhD challenge I had taken because we used to diagnose tuberculosis patients by taking their serum for aspergillosis antibodies. So what we were doing was we were preparing a 3-week culture filtrate of Aspergillus fumigatus, which is again a pulmonary pathogen, which causes very, very similar clinical symptoms as TB has. You have chronic cough, you have fever, and you have pulmonary picture. The X-ray radiology picture is completely akin to what TB produces, and there is the only difference is if you do repeated x-rays, you will come to know that the fungus produces fleeting shadows while mycobacterium has those shadows are fixed. So that is the only difference, but that is not possible everywhere and all the clinicians are not aware about it. So what we did was we after the genome sequence of Aspergillus fumigatus was revealed in UK, we sequenced our Indian Aspergillus fumigatus for the major allergen antigen, and this was with the help of NIH Bethesda. My mentor that time, Dr. Piyusha Sarma, traveled to NIH and got our antigen sequence, the purified antigen sequenced. And why we sequenced this antigen was that it showed antibodies in 85% of the cases. So this protein was called immunodominant antigen. And in addition, it was very important for the invasive form of infection because this protein had a property of ribotoxin. It was a ribonuclease, and therefore it was cytotoxic to human cells. And therefore, what we did was we mapped this whole 18 kilodalton antigen. We took help from Professor Kotekar's lab at All India Institute of Medical Sciences Biochemistry Department that time. She had developed certain softwares. In those days, I'm talking about 1998. So in those days, India had developed some softwares to predict epitopes, and I used that software. And those days were not internet days, so the softwares were in a floppy at Professor D.N. Rao's laboratory in AIIMS. And that's how we mapped around 10 epitopes, which were having T cell epitopes, B cell epitopes, as well as together T and B cell. And then the opportunity was, we were seeking an opportunity where to synthesize these epitopes. These days it is so simple. You just give it, outsource, get the peptide synthesized. But those were the days when these things were not available. So what we did was, we in our scientific advisory committee meeting, we got one of the experts who was from CDRI Lucknow, and he offered, after listening to our presentation, he offered, I can synthesize only one. You tell me which one out of these 10 is important. So I searched the literature, read whatever was possible for me in those library days, and I found that the N-terminal epitopes are supposed to be the most immunodominant ones, which will raise antibodies and which will also, prime the T cells. So we went for this 12 amino acid epitope, and he synthesized that for us. And then we went ahead to, formulate an immunodiagnostic kit. And I'm happy to share with you all that immunodiagnostic kit worked fantastic. And then the Department of Biotechnology, after a few years, provided us funds to develop this kit, manufacture this kit in GMP facility, and validate this immunodiagnostic kit. And this kit detects antibodies to Aspergillus. So the TB patients can be having Aspergillus co-infection. They can be only Aspergillus patients because of the misdiagnosis. Okay, so there are these two scenarios present. And of course, if they are Aspergillus negative, then you don't need to treat them with antifungal drugs. So most of the patients that used to come from Patel Chest Institute to my alma mater at CSIR IGIB in Delhi, we found that people were treated for 12 years with ADT and did not respond, and they were swelling the ranks of drug resistant TB. So that was the message that I had when I was working with it. So we standardized our diagnostic kit, we manufactured it, we validated it in 3 different hospitals, independent validation at PGI Chandigarh, at VPCI Delhi, and at RBI PMT, which is also known as RBTB in Delhi. So all 3 hospitals gave us fantastic information that 33% of the pulmonary tuberculosis cases, suspected pulmonary tuberculosis cases, were possessing higher antibody levels to both specific IgG and specific IgE to Aspergillus fumigatus. And that, took us to the next level. We have patented this kit. ICMR is very soon going to transfer the technology, and I'm hopeful that it will be a part of our TB program. So that was a story, inspiring story I wanted to share with you all. And I encourage you all to be a part of Indian Immunology Society because this is a very vibrant society. We are historically old. We are now 50-year-old. This is our Golden Jubilee year. And Professor Uma Kanga is organizing the Golden Jubilee Conference at AIIMS New Delhi, which is very near to you. And we, encourage you all to be a part because we are going to call all, aspects of stalwarts from all aspects of immunology in the meeting. And to tell you very frankly, the Immunology Day that we celebrate every year on 29th of April was actually proposed by Professor Stephen Kaufmann from Germany, Max Planck, and who is known for his research in tuberculosis. He has developed a new vaccine, which is called VPM1002, that is currently in clinical trials in Cape Town, South Africa, as well as in India. ICMR is conducting the trials with the household contacts of TB patients, and the results would be out soon. And Stephen was also there addressing us in the TB CME that we had at Miranda House on 3rd and 4th May. So Stephen proposed this, and it was accepted all over, and it happened in 2005. So Immunology Day celebration started from 2005. And another thing I wanted to tell you is Dr. Soumya Swaminathan. I hope I'm audible. Am I audible?
Yes, ma'am, you are audible.
Okay. Thanks. So Dr. Soumya Swaminathan, who was chief scientist at WHO and was formerly director general at ICMR and secretary DHR, she unraveled a great information that tuberculosis is endemic to our nation, not only because that we are susceptible to TB, because of genetic susceptibility or environmental susceptibility. She shared that 40% of the TB cases are actually malnourished. So the nutrition is the major cause of TB spread in our nation, and ICMR, National Institute of Nutrition, is taking steps towards this, and we will have special diets for those areas where TB is endemic, and that is one more strategy towards end TB goal by 2035. So I end my talk here. Thank you so much, Devinder, for this opportunity, and I'm so glad that I was able to address.
Thank you, ma'am. I think students, let's give a big big clap for ma'am, because you can see she's in the car She is traveling. Again, she has taken out time to interact with all of us. So thank you, ma'am. And we look forward to see you here at Amity soon. Yeah.
Yes, yes, grateful. And I would like to convey my thanks to Professor Narayan Rishi, who is, present here. And I see Uma also there. Yes, Uma is there. Yes.
Hello, Dr. Taruna. Thank you so
Hi,
much
Uma.
for joining us. And I also
Great.
want to thank you on behalf of Immunology Society. We are coming here, me and Dr. Agarwal, spreading the message of immunology on behalf of our society. And I will give them more details about society at the end of my talk. On your behalf,
Great. Yeah. Thank you, Umaji. I have sent the society form for students to Devinder. He should, send it by email or WhatsApp, whatever is possible for him. And I would like to thank Nishit also personally, because today was my booster dose for Nishit's lecture.
Yeah, I was looking forward to meeting you. I thought now, at least today, but
Yeah,
nevertheless.
Nishit, it is my vaccination for your talk completes. So it first dose, initial dose was at MH, and now my booster dose coming. So thank you so much.
Great.
Grateful.
Thank you. Thanks. Look forward to meeting you. Yeah.
Thank you, everyone.
Thank you, ma'am, for taking the time to conduct this engaging virtual session. Before we proceed further, we request Professor Narayan Rishi sir to felicitate Professor Nishit Agarwal sir. The next session will be conducted by Dr. Uma Kanga ma'am. Dr. Uma Kanga is currently working as an associate professor, Department of Transplant Immunology and Immunogenetics, AIMS, New Delhi. She has been awarded by the Transplantation Society, the European Federation of Immunogenetics, and the American Association of Immunology. She is Counselor, International HLA and Immunogenetics Workshops Council, Secretary, Indian Society for Histocompatibility and Immunogenetics, Vice President, Asia Pacific Histocompatibility and Immunogenetics Association, Chair, International Committee, American Society for Histocompatibility and Immunogenetics, and has also been Treasurer, Indian Immunology Society. She also has 70 publications in national and international peer-reviewed journals and has been awarded the Commonwealth Fellowship, Royal Free Hospital, London, 2008, as well as Fellow Japanese Society for Promotion of Science, 2014. We request you, ma'am, to expand our knowledge about impact on public health, disparity in transplantation need, and organ availability.
good evening. And first of all, thank you, Dr. Rishi sir and Dr. Devendra, for inviting me and giving me an opportunity, and also the Indian Immunology Society for allowing the Amity University to hold this WID session. And I thank my previous speaker for enlightening all the students here with a lot of science. I'm not going to teach you science here today. I'm going to take a different turn today because the idea of this WID was impact a little bit on public health. And I thought instead of science, there are other aspects that I would like to cover today of public health. So basically, I want you to be aware of the aspect that there is something called transplant immunology, which I think none of the universities in India are teaching. And there isn't any particular course on this. But we at the All India Institute of Medical Sciences do a lot of transplantation related activity. And there are many private centers in India that are conducting clinical services for transplantation. So there are big hassles how the transplant happens, and there are a lot of myths that people are not aware of. And you, as youngsters, I would just like to give you some glimpses at what happens when a transplant is required and what is the scenario in our country about the organ availability. So this is my institute where I did my PhD. I was trained by Professor Mehra, who established this specialty in India. And then I continued to work there as a scientist. I'm presently serving there as a faculty. And I hope to work till the end of my service tenure and retire from this institute, hopefully. So transplantation. So what brings to your mind when I say transplantation? Can any student tell me what do you think about transplantation? What comes to your mind? What has happened that one needs a transplantation? Okay, good. Anybody else aware of any other organs that need to be transplanted? Kidney, she already mentioned. Liver, heart, and I will show you several pictures here that several organs need and can be transplanted in human body. And not only organs, there can be a lot of tissues like cornea, tendon, heart walls, skin, veins, and bones can also be transplanted. So there are facilities available, and sometimes we are not even aware that these kind of transplantation do happen, and they are happening in our country too. Many of the organs. This is a life-saving and a very healing therapy. It is really life-saving for people who undergo transplantation because kidney transplantation, for example, if they fail, they have to be on dialysis. In the country like ours, where we are, mostly from large percentage from rural India, they cannot even afford a dialysis, which is twice a week, costing them 5 to 6 thousand rupees. So transplantation is one-time effort of spending money, getting a transplant done, and then they just have to do maintenance immunosuppression, which is still affordable by many in our country. But otherwise, it's a therapy which only rich could afford to if they had to. So what kind of transplants do happen? So just for the students, I would like to tell them that there is an autograft. Autograft means what? You transplant a tissue from one to oneself. Sometimes this is done with a surplus tissue or a tissue that can regenerate. For example, liver. You can give, a liver can regenerate. Skin is grafted, and again the skin is regenerated. What is an allograft? An allograft is a transplanted organ or a tissue from a genetically non-identical or an identical member of the same species. If it's an identical, we usually say that as an isograft, but if it is a non-identical but the same species, it's called an allograft. Of course, transplants can be done using animal organs. The work is going on. You must have heard recently in the last few months that there was a heart transplant done in America. It worked for a while, but there were a lot of gene modifications and other stuff need to be done. But it has still not reached the stage where it can be a commonly practiced therapy. But there is transplantation, and hope is there that one day we will be able to either generate animal organs in the labs or use transplantation to manage these patients who really need transplant. And there are split transplant. For example, I mentioned liver. The liver can be split and given, and the liver regenerates. So these are different forms of transplantation. And if you go through the international literature, what it would say is that most often organs are from deceased donors, but living persons can also donate organs. But in our country, it is the reverse. Most of our transplant program is the live related, and a very minor percentage is from the deceased donors. And what do you mean by deceased donor? Deceased donors means those patients who are declared brain dead or circulatory death. Initially, it used to be just brain death, but now circulatory death is also coming into the scene. So if anybody meets a road accident or for some reason is brain dead, and the family gives a consent or the patient has already consented, then the organ can be harvested and a lot of people, their lives can be saved. I mean, from one donor, you can imagine 2 kidneys, 1 liver, 1 heart, 2 corneas, hands, many tissues can be transplanted. So how many people can get saved from the life? And it's not a new thing. From early 90s, the work on transplants have been happening around the globe. And a lot of firsts are mentioned on this slide. You don't need to go through, but just imagine that from way back in early 90s, transplant activity had started. And in 1990, the Nobel Prize was awarded to these two gentlemen, Sir Joseph Murray, who had contributed and done the first organ transplant, and Sir Don Thomas, who had done the first hematopoietic stem cell transplant. So for cancers and blood disorders and thalassemias, the bone marrow transplant is done. And for today, I'm not going to talk on bone marrow, but just telling you about solid organ. And Joseph Murray was the first person to do a transplant in early 60s there, which was a successful human transplant, allo transplant done. So what is the status in India? Like incidence of end-stage renal disease where they would need transplant, there are millions of people who need transplant, and 75% are unable to take the benefit of the transplantation because it is either now they don't have affordability to reach a center, or they don't have the money to reach the center, or the centers don't have the facilities wherever they are. Liver diseases, 10th leading cause of death in India. Blindness, these are the major reasons why transplant is done and these organs are harvested. That's why I'm mentioning to you that lots and lots of blind people in our country, and they need to be able to see and experience the life the way we can, who can see. So if we look at the end-stage renal disease, what I was told by the nephrologist in my center, Dr. Agarwal, who's heading the department, that about every year 2.5 lakh people would require renal replacement therapy, and about 90,000 of them undergo dialysis. Either it may be peritoneal dialysis or it may be hemodialysis, but only about 8,000 of them would manage to get a transplant done somewhere or the other. The rest of them die without any appropriate treatment. So you can imagine how many lives need to be saved with this therapy of transplant. So what are the major factors that affect the transplant? Is the transplant has to be done. Firstly, there is genetic matching, which is very important. The better the genetic matching between patient and the donor, the better the survival. Also, many of our patients, because they undergo a lot of blood transfusions, sometimes due to pregnancy, sometimes due to infections, they develop antibodies. And because of these antibodies, what happens is the antibodies, the immune response kills the allograft. And that antibodies can be donor specific, and that we need to measure pre-transplant and post-transplant, which is a very expensive assay. But these are important factors that can affect the outcome of the transplant. And also, when the transplant is done, patient is given immunosuppression. Why immunosuppression? Because our body needs to develop tolerance to an organ. And when it takes time for us to develop a tolerance. So for the patient to develop a tolerance to an organ donor, it will take time. So until then, the body has to, immune system has to be kept under control to be immunosuppressed, so that gradually the body learns that this is self and tolerates the organ, and the organ survives. So there is a variability in every individual to absorb the immunosuppressive drugs, and many a time these drugs can also be metabolized and cleared very fast. So their genetic constitution is like that they will not retain the drug. And many times these drugs lead to a lot of side effects. For example, hypertension, malignancy, and other stuff. So there are very, very many factors that influence, and the major factors because of immunosuppression are nephrotoxicity, infections, and malignancy. So if there is a transplant and one has to jump this hurdle, the main thing that one needs to look at is how to control the rejection. So all the immunologists are working how to stop this rejection. Can we predict rejection? Do we have any biomarkers that can tell us this patient might undergo rejection? And a lot of work is going on to improve the organ survival. So the basic immunological barriers for transplantation are the HLA genes. So I would, because I am from a transplant immunology department, I would briefly touch upon the HLA genes that impact the transplant. Of course, there are other factors like the ABO blood groups. Previously, the blood group mismatch transplants were not done, but now the blood group anti-blood group titers can be measured, and if the titers are lower, ABO incompatible transplants are also done. And there are several other non-HLA antigens also that influence the outcome of a transplant, and today we are not going to discuss that. So what are the HLA genes? If you have gone through your immunology courses, you would know that these are the genes are located on short arm of chromosome 6. And we are here because HLA is here is what our statement, because it is the HLA that allows us that our immune system can control the pathogens. For example, our part of the world, there are a lot of, the environmental is that we are in a lot of pathogenic load. So because we have a diverse HLA profile, we are able to handle it. Had we not been diverse, we would not been able to. So the diversity in HLA actually protects us to generate a good immune response and then handle the pathogenic load. So what are these HLA genes? They are classified into three genes, three groups, the class 1, class 2, and class 3. Majorly for transplantation, we focus on the HLA class 1, the A, B, C on the extreme left, right that you will see, and the DR and the DQ in the class 2. These are really, really polymorphic genes. So at the moment, if you look at it, there are like more than 36,000 HLA alleles known. So we in the lab have to be able to have technologies where we can exactly identify the gene profile, HLA profile of the recipient and of the donor so that we can tell how much compatible they are. So the better the compatibility, better the matching. So this, and also very unique feature of these genes is that in this region, about 40% of the genes are immunologically relevant. They are functional immunologically relevant genes in the class 3 region as well. And that these genes are inherited en bloc. So it's not that one part of the short arm of chromosome 6 will be the MHC region would be inherited and not others. The whole gene complex is inherited en bloc. So that really is the unique feature of these, that these genes are in linkage disequilibrium. And this graph just shows you by over the number of years how many new alleles have been identified using different molecular methods. And currently, with the advent of next-gen sequencing, a lot of new genes and novel genes are being identified. So one lab needs to, when they are doing transplant activity, needs to be able to identify all of these. So these genes are expressed on cell surface, they are glycoproteins. And in every individual, because we have a pair of chromosome, one is maternal, one is paternal, we would have one pair of HLA-A, one pair of HLA-B. So one chromosome, the genes are expressed on the cell surface, and on the from the other chromosome, another set of genes are expressed on cell surface. So if you just want to look at a family, I'm just giving you an example. If this was the father and the mother HLA chromosome, the whole thing would be inherited en bloc. So one chromosome from father and one chromosome from mother. That's how we inherit. So we all of us have a 25% probability that we would share the same HLA inheritance from our parents, and a 25% of probability that we would be completely identical. So if one gets an A haplotype from father and the other child gets B, and one gets the C haplotype from mother and the other child gets D, then we would be completely identical. So if a transplant has to happen, it is also possible that the patient's own brother or a sister is a complete mismatch. There could be a half match, there could be a full match, or there could be a complete mismatch. So not that transplants are not happening with a mismatch. Transplants are happening, but we have to give a lot of immunosuppression. But bone marrow transplants cannot be done if there are mismatches. They are preferred to with a full match HLA donor. So how do we do the HLA typing in the families? When they come to us, we just look at the HLA genes and we look at all the siblings. And for example, if this third child who's a patient, what we would ideally look for is another sibling in the family who has inherited exactly the same 2 chromosomes from the parent, so that that can serve as a donor. But if it is not there, if the patient doesn't have a matching sibling, then alternate donors are accepted, maybe half match, maybe mismatch, or maybe an unrelated transplant. So how do these MHC genes play a role in generating the immune response? I think in India, we're all very fond of cricket, and my mentor taught us immunology, and he said, remember the cricket stadium and you would remember immunology. And he said that the MHC gene serves like a bat. अब आप क्रिकेट खेल रहे हो तो बैट तो अकेले कुछ काम कर नहीं सकता जब तक कि बॉलर बॉल नहीं फेंकेगा. So the ball is the pathogen. So once the pathogen comes, the MHC's role is that to present the antigen. So what MHC does, that the pathogen is broken into peptides, and the peptide is presented only by the MHC genes. So then once the MHC presents the peptide, then there is a fielder, and that is the T cell receptor. So the T cell receptor will look at the antigen or the pathogen or the peptide only in context of the MHC genes. So remember that this MHC immune response is a cricket stadium where you need a bat, ball, and a fielder. So there is a pathogen, there is the MHC, and there is a T cell receptor. And this is what happens in organ transplant, that when there is an MHC matching, there is an immune response is much lower, because the recipient thinks that immune system thinks that this is a self. So when you look at the transplant data from international centers, over the years, the transplant survival has improved. If you see this graph, improved not only because we have better HLA matching strategies, because we've also developed good immunosuppression, good technologies to do surgery, but the effect of MHC definitely comes up, that if you look at these graphs, if there is a zero mismatch between recipient and donor, the graft survival on the top is much better as compared to if you see the lower line, which is a 6 mismatch. So if there's a more mismatch increasing from 0 to 6, the survival is deteriorating in any set of recipient and donor, no matter what duration you see from 85 to 90s, and from 90s to 2000, and from 2000 to now. This is the scenario that HLA matching impacts the outcome of the transplant. Of course, similarly, when there are family donors, if there is a deceased donor who's an unrelated donor, then the graft outcome is not as good as when there is an identical HLA match brother or a sister. So now in India, what is happening? There are several centers, more than 270 transplant centers doing transplants, and it's all over India that the transplant centers are working. Despite all of this activity and all of these organs being transplanted, it is never sufficient. There is still a lot of gaps. Gaps because sometimes the patient don't have a blood group match donor. They don't have an incompatible, ABO incompatible donor also, and there is nobody unrelated who can give a transplant. And initially, the unrelated donor transplantation was not acceptable because the Indian law did not permit an unrelated transplant to happen. It was only in 1994 that the Indian government passed a law, and there was a gazette notification that said that yes, now we can accept the organ to be taken up from a brain dead individual, and that the organs can be harvested and transplanted to an individual who needs a transplant and does not have a matching family donor or any family donor in the family. So after this gazette notification was passed, the gazette stated that who can be a donor. The near relatives, the brother, sister, grandmother, father, mother can be donors. Then there could be other near relatives, for example, for a husband. Even initially it was not, but later on it was approved that the spouse can be also donor. The near relative, emotionally related individuals. So now spousal transplants are also happening. And also, the law says that if there is a foreigner who is serving as a donor, then there has to be a committee that is to review. Even for any other unrelated donor, there has to be a committee that reviews that there is no financial involvement, and this is a donation by choice. And then once the committee approves that this donor is fit, the transplants can happen. So there is a law and there are regulations. Without this, nobody can undertake a transplant. The centers has to have the approval to be able to do transplant, and all the regulations have to be followed. So in India, a lot of developments have happened. First kidney transplant, first multi-organ transplant, lung transplants, pancreas transplant. Many activities happened. First renal transplantation done at AIMS in 72. By now, we are doing around 150 to 200 transplants every month, every year. And many other centers in India are doing even much more than that, maybe a transplant every day. So they're even doing 360 transplants a year. And with the gazette notification and the law to be approved, this Human Transplantation Organ Act came up. So the cadaver transplantation, the brain death legislation was passed, and many deceased donor transplants are also happening. And with this, the country has developed a national organ and tissue transplant organization, which is run by the government of India. This is called NOTO. There are regional centers called ROTO. There are state-wide centers called SOTO. And all of these people register patients who don't have a family donor and need a brain dead donor to give them an organ. So these NOTO, ROTO, SOTO work together. And we have at AIMS an organ retrieval banking organization that helps register patients to be there, registered with them, and they're on the wait list. And when the organ is available, there is a deceased donor available, the organ is allocated by the NOTO to a particular hospital, and then the transplant is done. So if this is a few years old slide, but if you can look at the numbers that need a transplant and how many organs are available, you can imagine that there is a big discrepancy and there is a big crisis that the donors are not available. This has improved a bit over these years. This was a few years old newspaper clipping, but I think even now we have a big gap. What happened in our country was in down south in Tamil Nadu, there was a NGO was developed called the Mohan Foundation, and they are a multi-organ harvesting and networking association. They, in state of Tamil Nadu, divided the state into 3 regions and developed a networking program. And through that, they started allocating organs. And actually, in down south, the organ deceased donor transplant activity was very good. That's what taken as a model, and that's how the all over India the NOTO and ROTOs were developed. So this was actually the genesis was Tamil Nadu, where the organ donation activity from deceased donor happened. And let me tell you that when Jayalalitha was the chief minister there, Tamil Nadu state started off doing free transplants, and even now a lot of patients get a free treatment there. Transplant is free, immunosuppression is free, and that's how even people are willing to donate organs, and people are undergoing these transplantation. So we at Institute have this ORBO. they maintain the waiting list for the patients. And on this waiting list, the organ allocation is decided once the patient is registered over there. So people also fill up the consent forms and sign up for donation, that in case I sign up for donation and if I happen to meet an accident, I would be a volunteer donor registered, that if I am brain dead in the hospital, and I must inform my family, and if the doctors contact the family that Uma is brain dead and will not survive, then my family, if they are aware and my consent is there, my organs can be harvested and lives can be saved. So you all need to be aware that these activities are happening. I don't know how many of you knew that one could still register as a volunteer donor, that just in case I happen to be in a situation where I'm brain dead, I can donate organs. And these state-wise Noto Roto help all the organ transplants to happen. So this is just a data from the Noto website about the number of transplants that are happening, and you can see the majority of the big peaks are the kidney transplants. So in our country, majority of the focus is kidney transplant. Very few heart and liver transplants are happening at the moment. People are working towards improving this, and in next few slides, I will show you what is the activity happening. If you look at these bar diagram on the left, majority of the patients are males, very few females. And I must tell you from my experience, when the spousal transplants happened, in our institute, we experienced that it was always the female coming, wife coming and donating to husband, very rarely a husband coming and donating to the wife. And whenever we find a recipient donor pair where husband is donating, our lab celebrates. We are very happy and we say, wow, there is one donor, he has come forward for his wife. So it's improving, but you can see that there is a less, because I think the major focus is that the husband is probably, the male is the breadwinner in the family, so probably families also feel that this is not to be done. And also I must tell you that previously we used to feel a lot of patients who, when I was doing my PhD, I felt a lot of patients were coming forward and their brothers sisters were donating. But the moment the spousal transplant law was passed, the brothers sisters washed hands. The brother's wife will say, nahi aap nahi doge, sister's husband will say, aap nahi doge. And patients used to come and cry कि मेरे भाई बहनों ने भी मना कर दिया है. Parents used to come and cry. Parents are the only option because they will always do things whatever possible for their children. So spousal transplant has grown, but it is yet to reach a balance where males are becoming donors. So that imbalance is there. It's not only in deceased donor, live donor, everywhere is the same scenario. Even if somebody needs a transplant, probably we female in the family, families will not even take her as a donor. I see that when bone marrow transplant families comes to us with thalassemia, with cancer, when I ask them how many siblings, how many children you have, the parents will say 4 बच्चे हैं. 4 बच्चे हैं, बताइए बड़ा कौन है, ये बेटा है, ये बेटी का नाम ही नहीं लेते हैं. उसके बाद में पता लगता है कितने बच्चे हैं, 6. तो आपने तो बताया 4, तो बोलेंगे वो तो लड़की है. So this is how the attitude is in our country when people come from rural areas. So we really need to have a different mindset of patients and to get this benefit of transplantation. So this is the scenario. You see the pie chart, very few deceased donor transplants are happening in the country. It's majorly live related program at the moment. And on the Noto website, if anybody wants more education, there is information on FAQs are there on organ tissue donation, on transplant activity, what is happening, live related, deceased donor, legal, ethical matters. So if anybody is interested, can go and look at it. And they have wonderful key messages there. And I want you to read a few of them, that all major religions support organ and tissue donation. So please don't think any of your religion will ever stop you to donate organ. Religions do not tell us to discriminate. You may have not donated anything throughout your life. It happens, we don't donate, but can donate your tissues and organs after your death. So remember, if you have an opportunity to save a life, save a life. One organ donor can save up to 8 lives and enhance the lives of many others through tissue donation. So imagine 8 lives means 8 families. And how many people are impacted by one donation? One person donating, how many families are, and how many individuals in the community get the benefit? And why burn and bury your organs? Why not donate? I mean, we always either burn or bury. So we can change our attitude. And why destroy this gift of life? We have a gift of life, we can donate it. It's not causing us anything after death. What are we going to do with our organs? Nothing can be done, neither the family can get benefit. So organ donation is a way to continue life forever. All those people who have donated, when they come for celebrations of the organ donation day, we see that the families do cry and remember their loved ones, but they feel so proud that they were able to save lives. They cry and then they laugh. It's a very mixed feeling, but I have experienced myself, and I feel that when they come forward, they really think that the gift of life is to pass it on, and they do it. So pledge donate to donate and give people your hope. That is what my message is to you people. And how this organ transplant activity is happening. Now, initially it used to happen in one city. We were sending organs from one hospital to the other, but now it's happening all over India. You can fly organs. So people in PGI Chandigarh harvest organs. They fly from there. AIMS is ready with the operation theaters. Recipient, they start operating. The moment the flight lands, the doctor is coming with the organ. Even the police department is helping us. They clear the corridors, and then the transplant organs that is harvested is brought to the hospitals immediately. It's not only happening in Delhi, in many major cities in India. This activity has come up. People are now aware. Government is supporting. Traffic police is supporting, and organs are harvested. Airlines are supporting. Even drones are being now tried for transporting organs from one place to the other. So the activity is being promoted, and that is the deceased donor transplant. But there is another alternate to that. What is happening is that people in some centers have developed very nice strategy, and this is one center in Gujarat, the kidney transplant center. It was started by Dr. Trivedi. It's a beautiful transplant center where major focus is transplantation. They came up with an idea that we could swap donors. So there was a recipient donor pair, and there was another recipient donor pair. Their donors were not fitting with their recipient, but one pair donor was fitting with the second pair, and they could swap the donor. So they started doing this, and the swap donor transplant program is working very good. Many patients in the same center are benefiting from the swap donor. And it is not only swap from one patient to the other. People also do multiple. So there are one or two or three pairs. There is a closed chain donation. The first donor gives to second, second gives to third, third gives to first. And not only closed chain, there is also a dominoes chain that the Ahmedabad Center has done about 10 swap donors in a week, one after the other. 10 swap donors means 10 pairs. They could exchange the donor and fit one donor with the other patient, and they could get the benefit. So they didn't have to wait for deceased donor transplantation. They had a donor available in their families, but they were willing to give it to anybody else as long as they were getting an organ. So these are strategies for the shortfall, for the gaps that we are having for organ donation. So this domino effect is also there, that multiple individuals can get the benefit. But no matter what these activities are happening, there are still issues and challenges, and government tried to look at it, and they developed a committees, and they looked at that there is still huge burden of organ failure. There is poor availability of organs. The lack of awareness that this concept of brain stem death is not even there in the doctors. The doctors fight. The neurosurgeon says, why am I going to take the responsibility of declaring brain death? Why the other doctor is not taking the responsibility? So a lot of problems are happening, and people don't accept the fact of brain stem cell death. There's availability of infrastructure in the government sector is very limited. So all of these, there's a lot of reluctance in states. Some see the health is a state matter. Some states accept the central government law, some don't. So even the Organ Transplant Act is not accepted by some few states. So all of these problems are there, and considering all of this, what happened was that the government sat together again. They modified the organ transplant law, and they felt that why our country is not able to improve on organ donation. Our transplant rate is much lower. In a country like Spain, there is like the per million 48 per million are donating organs. In our country it's like point something. So why there is so much of discrepancy is because of the awareness, the infrastructure, and a lot of limitations. So government is working towards it, and they are trying to improve the brain stem cell, these brain death declarations. So these medical legal formalities, they're trying to reduce those, and so that they can improve this deceased donor transplant program. More transplant centers are being set up, and they are training, transplant coordinators, transplant activities in public hospital. Everything is being focused on so that this gap can be fulfilled. So this is regarding the major activities on the renal transplantation. So there is also another organ that people in India are majorly focusing on, and that is the heart and the liver. So I'll briefly give you a glimpse of the heart. The first heart transplant was done in our hospital in AIMS in India in 1994. I was a student. We remember we got a call at late in the night that there is an organ available, deceased donor available. The cross match had to be done. So normally the transplant immunology lab has to do a cross match between recipient and donor blood samples and say that the recipient serum does not have any antibodies to donor So if the cross match is negative, only then a transplant is done. So we did that cross matching, and Dr. Venugopal, our director, he was the one who did the first heart transplant in India. And that's when in 1994 he pushed and got this gazette notification also certified by, approved by the government. And 25 years down the line, this Mohan Foundation in down south, Dr. Shroff, wrote this article, Heart Transplantation in India, looking back. And what he mentioned that he looked at the global data, and he also found that even in the global data, they were like in 2017, 1,39,000 some thousand transplanted were done, but heart and lung was only about 10% of that. So the activity on heart and lung was pretty much lower. The major focus was on the kidney transplant only. And they felt that, we need to really push this activity. So what needs to be done? How do we go forward? And they looked at the literature as to when what transplants were, first transplants were done. And see, they pointed out in this paper that from early 90s till 2018, there were only few centers who were doing transplant, and these were the first doctors who did transplant in their states. And the transplant activity is limited to these states. It is not happening in all the centers in India. So it really needs a push. And they also compiled the data from, say, from 2012 to 2018. They looked at the data that the activity on the heart and lung, you see the lower blue bars, is not very high, whereas the red bar above is the kidney transplant. So activity is still focused on kidney. Probably that's where people feel that, that it can be easily managed or the organ will be easily available. Not many people donate hearts. So somehow this activity is to be promoted, and that's why people are compiling information. Where is the gap and what needs to be done? And they also in this paper compare data from the worldwide and look at the international data that there are thousands of transplants happening. And in India, the red line below is the Indian graph, that there is a gross, huge difference between the international setup and in our setup. And we really need to improve this, per million number of transplants or donation. So they calculated and mentioned that organ donation rate is like 0.67 per million in India. Of course, from in the last 5 to 10 years, it has doubled, but still, and for example, in the heart, average heart transplant rate is like 0.2 per million. Globally, it is 1.06. So we are way behind the globe in terms of heart transplantation. And in India, it has improved, like it was 0.01, it has come to 0.2, but globally, it has improved from 0.8 to 1.09. So people are moving at a fast pace, and we need to push this transplant activity in our country a lot, so that, if we create more awareness, probably more donations will happen. So they put down that in 2019, out of the 37 states and UTs in India, only 16 had, donation, the deceased donor transplant. And they mentioned that about 550 transplant units in about 250 hospitals are doing all kinds of organs, but less than 10% of them are in the public sector, in the government hospitals. So obviously you can imagine that if a patient has to go to a private hospital. I was just in a meeting last month in a heart failure society, and I asked the clinicians there, how much is the cost of a heart transplant or a lung transplant? They said the lung transplant costs about 60 lakhs. So you can imagine that during COVID, they did a lot of transplants in private sectors in down south. And imagine the people who could just afford to go for a transplant is 60 lakhs. I mean, I don't think any of us in this hall would, rarely few percentage might be able to afford, but it's really unaffordable. So if the government sector doesn't come in AIMS, we did the first lung transplant last year, and we've done just the third last week. And imagine that if AIMS is not able to do it, like AIMS, we have full developed heart transplant program from 1994. We have not been able to yet take off the lung transplant program so well. So if the government doesn't have, the government hospitals do not come up, at least in remote centers, what the government is pushing is that there should be an ability in the government sector to harvest the organs. So if they are harvesting, at least there can be few transplant centers, and if there can be a mechanism developed to transport the organs, at least some patients will benefit. So these are the limitations that our country is facing in terms of, promoting the transplant activity. So this paper mentioned that there should be a registry, and just last week, one paper has been published on Heart and Lung Society. Their registry, they mentioned that there are 70 centers in India who are doing transplants. I think it's a busy slide, but I need to tell you is that look at the states that they mentioned: Andhra Pradesh, Delhi, Karnataka, Kerala, Tamil Nadu, Telangana, and West Bengal. There are few states. Imagine how many states we have. these are the only states where the transplant activity is majorly happening. And if you look at this map, the red portion, nothing, no transplants happening over there. No transplant center doing heart lung. And they also try to ask the centers, please send us the reports, upload the information, how many transplants you have done. People are not even reporting, even doctors are not reporting what transplant activity they are doing. So we are not even able to judge what is the scenario. So everywhere, no matter the patient, the donor, the government, the doctors, everybody needs to join hands and push this activity. But they still compile the data that there are over 1,300 heart transplants done in our country, over 475 lung transplants done. I think this is information only from the government sector somewhere, but not from private. Private centers are doing a lot many more lung transplants as far as we know from the conferences that we go. Left ventricle assisted device are being implanted when patients can't go for transplant. 61 lung transplant data was there in this registry when it was published last year. So they mentioned that the hospitals have published individual data, but cost of the transplant in a government setup is 15 lakhs for a heart transplant. And this will vary from center to center. But once you spend 15 lakhs, the monthly immunosuppression cost is 30,000. Later on, it reduces to 15,000. So imagine how many of us can afford to take this immunosuppression post transplant. But we still have a lot of patients, transplant activity needs to be done. So the government needs to bring in private public partnership, somehow raise funding wherein that this money can be raised for the patients and they can go and get the transplant done. But they mentioned that the survival up to 1 year is about 80 to 90% in most centers. For lung transplant, one published series shows 3 year survival is 76%. So it's really worth it if a patient can be offered an organ and can be transplanted. And they concluded that in India, is doing about 90 to 100 transplants a year, and there are out of the 70 centers, 26 have done more than 10 transplants. So there is indication that some centers are very experienced. And the paper also says that what is the way forward? What do we need to do? We need to strengthen the national program. South India and West India are doing very good in terms of active brain dead donations and doing, but rest of the India is not as well. We need to establish Indian Heart and Transplant Registry. When this paper was published in 2020, registry wasn't there, but I just showed you data from the registry that was developed in 2 years. Data is there. So we need to follow how many transplants are being done, what is the short-term, long-term outcome, whether they are successful or not. We need to increase the organ donation. So retrieval centers need to be developed. Then donation after circulatory death. Instead of brain death, circulatory death is also now considered that they can donate organs. So people need to accept that circulatory death and donate organs. So creating cross subsidy through public-private partnerships. That is what the government is also trying to now push. If the private sector can come in and support the public sector. Improving regulations. Sometimes even the policies are made and the implementation is delayed and there is no good policies are made by the government, it's not implemented. Cost of post-transplant immunosuppression needs to be brought down. Government can import drugs in bulk, provide to the patient. Then they can also set up the core transplant facilities, core center for excellence for transplantation, and also develop mechanism how the organs can be transported from one center to the other. This is another paper from our center. The two cardiac surgeons were trying to evaluate what can be done and what are the challenges for thoracic organ transplantation in government institutions. And the major factor they said was the financial implications, infrastructure and patient profile. The patients are not in a situation to pay for money, and their clinical condition is also not good. They reach the tertiary centers when they're really in a very bad state, even they cannot be offered a transplant. Had they come on timely reported to the center, transplant could be offered. So harvesting organ and the logistics of transport, trained manpower, training the nurses, training the residents, training the doctors, the transplant coordinators. Everybody needs to be aware. Even now, I can tell you the transplant immunology labs, when they get a call at night that there is an organ available, you have to go and do a cross match. Sometimes my own center, the doctors, the scientists will say, oh my God, now middle of the night, you're sending me to do a cross match. So I just tell them, imagine you are spending 3 hours, you're going to save life for somebody. So you have to push. Sometimes it really takes a toll. And I can imagine there's no incentive to that. They're not given a transport to come. They're not paid extra money. Nobody appreciates them. They don't get a promotion. They don't get any extra salary. So obviously they also feel, why should I do all this work? But for the community, as human beings, we have some responsibility, and this is what we need to do. So there are unmet goals. That is, the government policies are formed, and they are not implemented due to certain regulations. It takes approvals by the government, and then those policies sometimes need to be changed before they're implemented. So that's the scenario of organ transplant, and I wish to thank you all. And I want to tell you that if you can save a life, life is amazing, and you can pass it on. I mean, our organs can live in somebody else, and we can pass it on. And now, as member of the Indian Immunology Society, it's my responsibility to also tell you that the society is there, existing for last 50 years. From 1702, it was established in Department of Biochemistry at AIIMS. We have about 1,500 members in the society who are active. There are a few who are not active. And the society is doing a lot of good for spreading the message of immunology. For example, we do a lot of CMEs, workshops. Society sponsors them. We do International Day of Immunology like we have done today. We do annual scientific conferences, which we name as Immunocons. Every year an Immunocon is done. And during the Immunocons, a lot of young scientists, mid-career scientists, senior scientists, everybody gets an opportunity to apply for awards. Young budding scientists who are below 35 years can apply. Their research work can be submitted. They can be considered for Professor G.P. Talwar named Young Scientist Award. Professor Talwar is the pioneer. He is 96. He is the one who established the society in India, and he is very active now. Even now he comes to the conferences, and he's first one to enter the hall, and he sits in the front desk, and he listens to all the discussions, and he also delivers talks. He also travels. So it's a really an amazing personality to meet. And on his name, he started Young Scientist Award, and then he also initiated the Mid-Career Scientist Award. So people who are 45, they can apply for a Mid-Career Scientist Award, and others who are senior can apply for a Senior Scientist Award. So society gives one award, one senior scientist, one mid-career, and 2 or 3 young scientist awards. So if you take up immunology as your main research area, sometime in future you may be able to, but you need to be a member of the society to be able to apply. So join the society if you want to gain more on immunology activities in the country. So as Dr. Taruna mentioned, the membership forms are available. It has been emailed to Dr. Devender, and he will email every one of you if you're interested. Join the society. You can be a life member. Once you pay, you become a life member. And I welcome all of you to AIIMS. Because of the 50 years completion of the society, we are going to have a Golden Jubilee celebrations at AIIMS. And this will be a big scientific meeting in October. So you note down your date, October 5 to 8. And all of you are welcome to join the conference. There will be opportunities for young scientists where they don't have to pay registration fee if their papers submitted are good. So I encourage all the master students or the PhD students who wish to join and can get some opportunity. And if you volunteer and help us organize the conference, even then your registration fee will be exempted. So I encourage you to be a volunteer and also be a researcher. Join the conference. We are going to invite a lot of speakers, and you will get opportunity to listen. Today you heard Dr. Agarwal, one tuberculosis theme. There will be a full session on infectious disease, and you will listen to him and maybe many more. And you can imagine how much you will gain. So you heard 1 hour, and you must have gained a lot on CRISPR-Cas. I learned from your talk, although I was doing slides. Sorry for that, but I learned too. Wonderful talk. And I think the students will gain a lot during the conference. And for today, I want to thank Professor Rishi and Dr. Devender for organizing this event and giving me an opportunity to just tell the student what is transplantation. So no science. I'm sorry, I didn't talk any science, but just for you to know that what this transplantation activity and where we are. Thank you for your time, and I will appreciate if you all join the Indian Immunology Society and also be a presenter sometime in our meetings. Thank you, Taruna, for staying back and listening to the talk. Yeah, wow, thank you. If anybody has any questions, yes, go ahead. You first. Okay.
So actually, ma'am, we know that there can be occurrence of mutation in HLA genes. So I was just wondering that can transplantation still occur if there is mutation in HLA genes?
Yeah. See, the transplantation can happen even without a mismatch, without a match. So of course the match is preferred. The survival, long-term survival is much better if you have a better HLA matching. But if there is a mutation and there is no matching donor available, with a mismatch also transplant is done, but the patient has to be given heavy immunosuppression, and then there are side effects of the immunosuppression.
Ma'am, but sometimes what occurs is if there is mutation in HLA genes, the recipient when receives the transplant,
Yeah.
it can also perceive the organ as a self antigen or an foreign antigen.
Yeah.
So, ma'am, there would be chances of failure. So, how will you evaluate that?
So there won't be mutation because of the transplant.
Ma'am, not because of the transplant,
Yeah.
but because of the mutation, transplantation failure can occur. So,
Because of mutation, transplant.
ma'am, as in the organ would be by the HLA mutation. Sometimes what happens is the body recognizes the organ which we are receiving as in as in foreign antigen. So, ma'am,
See, the organ is anyway foreign. Problem happens when body recognizes foreign as self, and that is when autoimmunity happens. So if the immune system, the immune system thymus is like your MIT university where you get your education. When cells go to thymus, they get educated. What they learn over there in thymus? They learn how to discriminate self versus non-self. So if there is a self-reacting cell that gets deleted, there is clonal deletion. And if there is a cell that can react to foreign, that is what is retained in the body. That is how immune system learns. This is the education that the immune cells get when they pass through the thymus. So our body is tuned to recognize self and not react, and tuned to recognize non-self and react to that. So organ, when it comes, body will think it is a non-self and react. But if the organ is genetically matched, then body will think, "Arey, this HLA is the same as my own HLA, so why react?" The HLA will not present the antigen. So there will be less alloreactivity. And that less alloreactivity will lead to more tolerance. And the organ will be accepted. Only when there is non-self, the body reacts. So mutation hoga, agar mutation hoga, so anybody's HLA genes are mutated, if that kidney is given, obviously it will be non-self, body will react. But then immunosuppression will control it.
thank you, ma'am.
Yes.
It was an interesting talk, ma'am.
Thank you.
My question is that um, if a person happens to get an organ transplant done, and unfortunately, if that person is a latent TB subject, so knowing the opportunistic nature of the pathogen, Mycobacterium tuberculosis,
Yeah.
he or she should not be given immunosuppressants.
Yeah.
So, what should be done for such patients?
So the patients, recipients are evaluated before they are taken up for transplants. So such patients will not be fit for transplant category. So there is a clinical evaluation criteria, and only if the patient is fit to undergo transplantation can they be offered. So obviously, if they are diagnosed as tuberculosis, they would first be treated for management of tuberculosis, and when they are tuberculosis is treatable.
Yeah. But if it
So-
is a latent TB infection,
Even if it is latent, so they are investigated, all the investigations that he said before the transplant, all the gold standard and the newer ones, everything will be investigated. So the fitness of the recipient and the fitness of the donor is evaluated prior to transplant on the function of the kidney and on general fitness, and only then a transplant is done. So obviously the patient is sick, his kidney is not working, kidney has failed, he is undergoing dialysis, he has multiple infections. So for infections, there is a control, antibiotics are given. Of course, if it is tuberculosis and it is not diagnosed at the time of transplant, then the patient will go for transplant. But if it happens post-transplant, there will be a balance between the immunosuppression and the tuberculosis. The patient will have to bear with tuberculosis till the organ develops tolerance. When the immunosuppression is chilled, anti-TT will be given. So that's the clinical scenario the clinicians will manage at that time. But usually they are screened prior to transplant. Very well screened.
thank you, ma'am.
So she put the two talks together, the tuberculosis and immunology. So I think you were not sleeping. I just recently was in one another WID, and one of the speakers from ISC Bangalore, Dr. Nandi, was talking, and he, in the middle of the talk, he put up a slide, and he put a young gentleman with big muscle, and he said, "Body to bana liya, antibody nahi banaya." And then he said, "I put up this slide because it's a wake-up slide कि सो ना जाए बच्चे." तो I think ये सो ही नहीं थी. इसने tuberculosis भी और organ transplant भी सुन लिया. Good.
Hello, ma'am.
Yeah. Hello.
Ma'am, I have seen that there is the transplantation cases for heart and kidney. It's improving, and there is also thoracic organ transplantation in the pipeline.
So, ma'am, what is your projection that when can we expect brain transplantation to happen?
Oh, brain transplantation is a big thing. I cannot imagine, cannot predict that at the moment because we are still fighting with the main organs, and it will take a while. I think so. It's not. But then, why would we need brain transplant? Because if the brain has problems like gliomas and other things, those are surgically treated. So if those can be treated, then we probably wouldn't need a transplant. So it all depends on if the brain is completely gone, then the transplant would be required. But if it is treatable, neurosurgery has improved a lot in the globe, and majority of the tumors and everything can be resected. The patient can be given chemotherapy, radiotherapy, and managed. Yeah,
Okay.
hard to predict. Anybody else? Others were sleeping or awake?
It's an excellent talk. It was very engaging. I don't think anybody would have slept actually. So I was wondering with the statistics which you have shown between Western countries and the India, like why do you think that? Where is the problem actually? That huge gap actually.
The problem is majorly because of the awareness of organ donation and also about our beliefs. In our country, we just don't believe that we don't need to take our organs to heaven. We face a lot of this problem that when our neurosurgeons tell us that whenever there is a deceased donor, like road accident death is so common in our country.
Exactly.
And there are so many patients who come to our trauma center, but very small percentage of them do give consent. Even if the law says if the person has consented, even then the family consent is also to be required. And many a times people give consent and they don't inform their families. So it's our responsibility that if we give a consent, we should also inform our families that if I happen to be in that situation, give. So that is the major factor that people don't come forward for donation. Somehow there is a myth that body ko kharab ho jayega, kaat denge, nikal denge organ. That kind of feeling is there, and we are not coming out of it. And government is trying, but there are not awareness programs also.
Exactly.
Like for bone marrow transplant, let me tell you, we have been working since 95 to develop a bone marrow registry in India. We don't have a government set up a registry. I have been going to so many camps and blood donation camps and teaching them कि in bone marrow donation, you know what happens? We just have to give little bit blood. Like जब dengue हो जाता है, हमें platelets देने होते हैं. So we are just giving blood. And in that blood, the donor is given just one injection and the stem cells mobilize from bone marrow to blood. And we only have to give blood. So you hook on the machine, इधर से ब्लड जाता है, स्टेम सेल सेपरेट होते हैं, उधर से ब्लड वापस आ जाता है. 2 घंटे मशीन पे बैठना होता है, and all of us can be donors for stem cell. If everybody's HLA is tested in the computer, any patient needs, I can just screen the computer, anybody is matching, call them and give. उसके लिए भी नहीं मानते हैं. Leave aside that, hematology doctors, oncology doctors who are treating these patients, all the DM students come to me for lectures on transplant immunology. If I tell them register for bone marrow registry, they don't register. What to tell about general public? None of the physicians in my center are registered as a volunteer donor for this thing. So awareness is a big problem. And then also, I think the infrastructure there is much better. Probably because I think in the other countries there were smaller families and family donations were not happening. So they developed this mechanism. In our country, initially the family bonding was there and families were supporting, so that was happening. And also because of our myths, awareness. और अब जब awareness हो रही है, तो इतने infrastructure नहीं है, इतने trained physicians नहीं है, nurses नहीं है.
So, like the rate of success, is it same here as well as?
Pretty good. We are organ donor, organ recipient, older one. I think the longest is about 35 years old transplant.
Wow.
Yeah. The first heart transplant that was done, Dr. Venugopal, that patient survived 14 years. We have a heart transplant patient who's working in Orbo in our institute.
Very good.
And survival is pretty good. And lung transplant also, I have Heart and Lung Failure Society meetings I went. People say that their patients are doing very well. So imagine if 70-75% patient also survive, that is really good.
That's true.
Pretty good. Survival is good. It's not bad. Only thing is those people who are non-compliant will not take immunosuppression. Then obviously they'll, organ will fail. That is the thing.
So I think more questions can be taken during the tea.
Okay.
So
Thank you.
Yeah.
Thank you, ma'am, for this insightful session. We now request Professor Nishit Agarwal, sir, to felicitate Dr. Uma Kanga, ma'am. This event would not have been possible without the meticulous efforts of Dr. Devinder Toor, sir, the organizing secretary of this event. Dr. Devinder Toor is presently working as Assistant Professor 3 at Amity Institute of Virology and Immunology. He did his bachelor's in microbiology from University of Delhi, master's in medical biotechnology and PhD from PGIMER Chandigarh. He has more than 10 years of teaching and research experience. At Amity University, he has been awarded 8 research projects, including 2 international projects in collaboration with University of Utah, USA, and Indiana University, USA. His major areas of research include the study of pathogenesis and genetic susceptibility markers of rheumatic heart disease. His other areas of research include surveillance of antibiotic-resistant bacteria from clinical and environmental samples. With this brief introduction, we welcome Dr. Devinder Toor, sir, to deliver the vote of thanks.
So, good evening to all the dignitaries, my fellow colleagues sitting over here, participants, and my dear students. As all good things come to an end, so does this small event that we organized. And it gives me immense pleasure to extend a vote of thanks for this event. As organizing secretary of this event, I wish to state that around 200 students registered for this event from different universities of Delhi NCR. I'm very much sure and I'm very confident that this symposium and the wonderful talks by the speakers must have inspired you towards this exciting field of immunology. And today, I think we have heard about 2 major subjects, transplant immunology and the tuberculosis research that is going on. Plus, Dr. Taruna is there. She's also, teaching reproductive immunology. So, you have heard about a lot of different subjects, and Dr. Taruna will surely will be here and talking more about the subject of reproductive immunology with us. So, I think this event surely has been successful in achieving its aim and has successfully motivated the young minds and inspiring them towards this exciting field of immunology. So just starting with the thanks, firstly I wish to thank all the eminent speakers for sparing their time from the busy schedule and sharing the research in the field of tuberculosis and transplantation. We are really enlightened and very happy with your presence here, and all the 3 speakers have been very supportive and helpful to me. Also, students, I wish to tell you that our students have done dissertation with both Dr. Nishit and Dr. Uma, so you can always approach them for your dissertations later on. Next, I think the words are less to thank Professor Narayan Rishi, advisor and founder of Amity Institute of Virology and Immunology. He has been the pillar around which our institute has flourished. So he has taken all the pain for getting the things done for this event. So thank you, sir, for all your support. I have no words to express my gratitude for our beloved Honorable Founder President, sir, Honorable Vice Chancellor, Madam, and Professor W. Selvamuthi for their continuous support and blessing and for showing us the path to excel. I also wish to thank the faculty members of AIVI. There are some of them are sitting here and at the back. So I want to thank them who have always chipped in with their constant inputs for the event and extending their support to me at every time. And special thanks to the participants for your enthusiasm and patient hearing. And I'm sure you will again catch hold of the speakers outside while having the tea. So it was heartening to see the young minds getting motivated and inspired in this field. A very big thanks to the Indian Immunology Society for supporting and funding this event, without which this event would not have been possible. I also thank the IT team of Amity University for all the setup they have created. And
We would once again like to express our gratitude to the distinguished speakers of this event for taking time out of their hectic schedules to broaden our horizons and shed light on the important aspects of immunology. We are also extremely grateful to Professor Narayan Rishi sir and Dr. Devinder Tur sir for organizing this event, as well as to the Indian Immunology Society for sponsoring the event. Now, we kindly request everyone present here to head outside the hall for high tea. All attendees will be receiving an e-certificate. We once again thank you for joining us today and making this event